Hello, everybody, and welcome to day 2 of the Genomic Medicine 13 meeting. We had a really productive day 1, a lot of input and interactivity despite the virtual format, so we're hoping that that will continue today. Let me just provide a little bit of orientation for how day 2 will work. We will have 2 sessions that are going to be very similar to the sessions that we had on day 1 with different areas of content focus. We're then going to be taking a longer break, approximately 1 hour towards the end of the day today. That will allow my co-chair, Ken Wiley, and myself to synthesize our takeaways from day 2 and merge them with some work that we've already put in for takeaways for day 1. And then we'll reconvene the meeting for an open discussion where we'll walk through the takeaways to make sure that we captured the information accurately, and also to make sure that we did not miss anything that was important. This session is going to be really critical for our next steps, which will be to create a meeting summary, but also to develop a publication that will let a wider audience know what we have been trying to accomplish. And of course, the major takeaway will be for this meeting to inform work within the NHGRI about developing a research strategy, research portfolio around clinical informatics and genomics. And so this will hopefully result in the next few months in some information from NHGRI about how we're going to move this forward. So with that as the prelude, first of all, I want to begin by thanking all of the presenters yesterday for being very attentive to time limits. We hope that that will continue today. We want to thank all the co-moderators for the excellent job that you've been doing in terms of moderating the discussions after the presentations. A reminder that you can contribute questions either through the question and answer box, through the chat function, or verbally by letting the co-moderators know that you have something to contribute. All of those will be captured. A reminder that we are recording the session, so all of this will be recorded and will be ultimately available on the Genomic Medicine meeting site, where all of the prior meetings reside as durable materials. These are happening long enough now that some of us who have been involved from the first meeting are beginning to recognize changes in appearance over the course of the years that have taken place. And unfortunately, it's not a Dorian Gray experience for most of us. So in any case, let's go ahead and launch into session 4, defining a research agenda that addresses the process for developing genomic-based clinical informatics resources. This session will be co-moderated by Dr. Erin Krauge, who is the Director of Medical Bioinformatics at Nemours DuPont Hospital for Children, and is also the Director of Management of the Delaware ACEL, which is the Accelerating Clinical and Translational Research for Delaware. Dr. Krauge has a broad research portfolio with the goal of implementing a learning healthcare system to enhance precision medicine. Our second co-moderator for this session is Dr. Dan Roden. Dan is a member of the Genomic Medicine Working Group, has been from the inception of this group. He has a very long history of research in genomics and personalized and precision medicine, and has led programs such as the Vanderbilt BioView program, the Predict Pharmacogenetics program, and has been PI on projects through the Pharmacogenomics Research Network, the Electronic Medical Records and Genomics from phase 1 through phase 4, and has co-PI of the All of Us Data and Research Center that's housed at Vanderbilt. So with that, I will turn it over to Drs. Krauge and Roden.
Thanks, Mark. Am I muted? I'm unmuted. So thanks, Mark. Erin and I have divided the work up so that I get to do very little work and she gets to do a lot of work. I'll be introducing the speakers and she'll be taking notes so that we can summarize at the end and we can send our notes on to Mark and Ken so that they can summarize for all of us. So our first speaker is Teresa Caban, who is in the Office of the National Coordinator and in charge of personalized medicine as Chief Scientific Officer. So the title of her talk is Advancing Health IT to Support Genomic Medicine and Research. I'm going to put on a 9-minute timer, Teresa, and I will yell at you when it's 9 minutes. I will gently say that it's 9 minutes.
Sounds good. Thank you. Dan, as Dan said, I'm Teresa Caban. I'm Chief Scientist at ONC. Next slide, please. And before we dig in, I actually wanted to put some of our work at ONC in a little bit of context because I know that we collaborate quite a bit with NIH, but realize that not all of you may be familiar with kind of our space. And this is an attempt to try to show the Venn diagrams of within healthcare where we sit. As an office within the Office of the Secretary of the U.S. Department of Health and Human Services, we have two primary responsibilities with regards to health IT. One of them is that we coordinate policy efforts around health IT across the federal government and private sector stakeholders, but we also regulate health IT systems. So there was a lot of conversation yesterday about health information technology and how it may facilitate or hinder genomic medicine and research. And we have a role in that we certify health IT systems and their functionality. And most recently, through our 21st Century Cures Act rule, which was released last year, we have required that EHR systems use a standardized application programming interface, as well as a standard set of data elements known as the U.S. Core Data for Interoperability, both of which I think will be very important for research. Next slide, please. My division in particular focuses on putting health IT to work for research. While we don't fund research per se, we collaborate very closely with those who do, NIH being one of the agencies we collaborate and coordinate with. We also lead demonstration projects that try to advance technology and policy issues to be able to advance the research agenda. And the 21st Century Cures Act really prioritized interoperability, as does our regulation. And as I mentioned, it's pretty critical to, now that we have these health IT systems in place across the country, make them work not just for clinical care, but also for research and other use cases such as post-market surveillance and public health. And we have a role to play in advancing that. Next slide, please. And so what we have found through our demonstration projects and our coordination is just what a critical role health IT plays in the research enterprise. Being able to identify research participants, assemble cohorts, collect and share data, as well as share results back with both participants, clinicians, as well as caregivers and policymakers requires a robust infrastructure to do so. And what we have been focused on through our projects under not only precision medicine, but also patient-centered outcomes research, our LEAP in Health IT funding opportunity, and some more recent efforts to advance standards specifically, is in finding a way to do so in collaboration with a lot of different stakeholders, researchers and research funders included. Next slide. as I took over as chief scientist, I wanted us to take a step back and, much like what NHGRI did over the last year or so, think through what it is that is most critical to be able to leverage health IT and data from health IT systems in research. We led a multi-year stakeholder-driven effort to try to ascertain what those needs might be and articulated this in an agenda that was published early last year. We really are trying to foster an ecosystem where clinical care and research come closer together. And as you'll see on the next slide, the agenda has two sets of priorities, one focused on leveraging data and the other one on building the infrastructure or advancing the infrastructure that we need to do so. I'm not going to delve into these in detail today, but I encourage you to take a look. I'll say that a lot of these align with some of the priorities that NIH has broadly with regards to data science and informatics, and we were fortunate to be able to collaborate with not just NHGRI, but the National Library of Medicine, the Office of the Director, all of us, and NCI on pulling this together, as well as colleagues from the Food and Drug Administration, the Centers for Disease Control and Prevention, and the Veterans Health Administration, and a lot of private sector stakeholders. So through a lit review, a workshop, many conference sessions, we gathered a lot of input on feedback on pulling this together and are starting to work on moving forward on some of these priorities. Next slide. I wanted to dive a little bit deeper, though, into some of the work that we've been doing under the Precision Medicine Initiative, which, as you may recall, was launched under the Obama administration in 2015, with the goal to really move us to deliver care that is tailored to patients as individuals. And while genomics has a big role to play in that, we've been conscious from the start to focus on other things like environment and lifestyle and how that comes together with biology. When initially launched, the Precision Medicine Initiative included the National Institutes of Health, or what became known as the Olivis Research Program, NCI, the Food and Drug Administration, and ONC. The administration recognized the importance of infrastructure issues and the criticality of that to be able to advance science and discovery. And so since then, we've had a whole host of partners from across the federal government, the Office for Civil Rights, the Department of Energy, the VA, DOD, NIST, and I'm sure I'm forgetting others. And so collaboratively, we've been advancing sort of the puzzle pieces to be able to make this a reality, initially through discovery and sort of building the infrastructure to advance the science. Next slide. And so our role has really been consistent with what I said earlier, focusing on some of the policy and technology issues that need to be addressed as they pertain to health IT and electronic health data that comes from these systems that are needed to be able to conduct precision medicine research, as well as deliver precision medicine care. And we've done that through we've collaborated on some policy pieces, developing privacy and security principles and guidelines, as well as testing those through the Sync for Science pilot project, and advancing the use of standards to promote data sharing, as well as sharing specific data types that are needed for precision medicine. So in addition to collaborating with NIH, Harvard, and leading developers in Sync for Science, which is meant to leverage an open API to enable patients to share their data from their doctors' EHR systems in research, we've also been advancing standards for social determinants of health and mobiles and wearables. And then, as you'll see on the next slide, we've taken a phased approach in collaboration with colleagues who are joining us today and joined us yesterday to advance the sharing of genomic information in a standardized way. So Sync for Genes is an ongoing project, and we've been very proud to have been leading this in collaboration with many different stakeholders. We've been leveraging the FHIR standard, which is now required under regulation, and testing it, sort of seeing real world how easy or difficult it is to advance genomic data and share it in a standardized way. So initially, we kind of tested out the specification and built it out. In phase 2, we looked at whether settings could actually leverage the standard and specification to exchange data. And phase 3, which concluded last year, we started to engage laboratories. And phase 4, which just launched, and we've been identifying sites to do the testing, we will be focusing on sharing the data with patients and caregivers. For all of these, the one thing I wanted to emphasize is not only the importance of engaging organizations that are going to be leveraging these standards and collaborating with them, but also the standards development organizations to make sure that the results get integrated into sort of the standards development lifecycle and support broader adoption. We've been lucky to collaborate with Dr. Freemuth, who's joined us today in this effort, and it's really helped to spur collaboration. Next slide, please. And so before I conclude, I wanted to sort of throw out some questions to help foster conversation and help NHGRI. I think as the Institute considers developing this informatics agenda, it's important to be crystal clear in terms of what's within NHGRI's sphere of control. Is it the research piece? Is it the informatics for research? Is it broader than that? Is it clinical care? And then as we're thinking of informatics for who and for what purpose, right? Who is to benefit? Is it the researchers, the clinicians, patients, participants? And what are the specific data and informatics needs? I think we have a unique opportunity to try to think through what those needs are and build systems that support those instead of developing systems and trying to make sure that they fit a need that we have conceived. And then how to best engage other stakeholders. And for example, what role can ONC play to help advance informatics needs that are identified today and that were identified yesterday? And how can NHGRI engage stakeholders better? And so with that, on the next slide, I included a few resources and links that I think will be of benefit. The ONC Cures Act Final Rule, the link to USCDI. I want to point out this was released as a draft last year. The draft version 2 was released last month, and it will be shared with ONC's advisory committee, the Health IT Advisory Committee, or HIPAC, through a public comment process. And so I really want to encourage this community to engage in this process more fully, because as you identify needs for data elements that could be added or incorporated into USCDI, there is an opportunity for you to provide public comments as this standard gets updated on an annual basis. And with that, I'll conclude. Thank you.
Well, you're on time.
Excellent talk, Teresa. There is one question that came through the chat box. I don't see anything genomics related in any stage of USCDI. Is it proposed in USCDI somewhere?
Not that I've seen, no. Right now, the data classes and data elements are for those that are currently within the EHR. So I think if that's something that this community would like to see move forward in either version 2 or version 3, which will open for public comment at later this year, there's an opportunity to do so. Part of the issue with what gets incorporated to the USCDI is the maturity level of a given standard, and there being sort of a broad need for a particular use case, right? Because USCDI gets incorporated as a requirement of developers. So ONC tries to make sure that both the need as well as the technology and the standard are at a point where it makes sense to put that requirement forth.
So you got a real quick absolutely in terms of incorporating, and perhaps a group like Sync4Genes project can propose it.
Well, the Sync4Genes project is an ONC project, so it'd be a little awkward for us to propose a data class or data element to ourselves, but certainly we create a community. And so folks who participate are more than welcome to suggest that through the public comment process. And I believe NIH is also setting up an internal to NIH process by which the institutes and centers will provide formal comments to ONC on a go-forward basis.
Great. I don't see any other questions coming in. I have just one question. I was wondering if you could highlight or discuss potentially the importance in terms of consuming electronic healthcare record data for research, common data models, and the challenge that institutions, face with kind of transforming their EHR data into a common data model.
Certainly, I think that's an issue that we recognize at ONC and our colleagues at NIH. I had an opportunity to work at NIH under a detail for a year and was coordinating efforts to advance the use of FHIR in research. And something that we're looking at very closely is how the FHIR specification aligns or doesn't with common data models used in research, and what tools might be needed to be able to enable consumption of the data and maybe some transformations to facilitate that.
Fantastic, that would be great. Is there any more questions? Give a second here to see if people are typing.
Does your brief include engaging patients, engaging individuals who actually might be the recipients of genomic information, and how that should be delivered, whether it's sort of through their smartphones, and access to their electronic records? How do you not only provide access, but provide sort of an interpretive commentary along with the access to data? It's hard to think of data as just being dumped on the participant.
Certainly, and to sort of take a little step back with regards to your question, I think that, you know, individuals should have access to their clinical data, period. But the value to be derived is in what you do with the data. Yeah, what you do with the data and how you leverage that information. With regards to Sync4Gene specifically, that's something we are looking at now, and certainly through prior phases, what we found is that we need additional resources for patients and caregivers, as well as clinicians, when genomic information are shared.
If there are no other questions, I think we'll we'll move on. The next speaker is Subha Madhavan, who will talk about genomics-based clinical informatics resources to support precision oncology and evidence curation. Subha is the Chief Data Scientist at Georgetown and the founding director of the Georgetown Innovation Center for Biomedical Informatics. She's done an extensive amount of work, particularly in the oncology space with Georgetown and with MedStar, their healthcare partner. Take it away, Subha.
Great, thank you so much, Dan. Can everyone hear me okay?
Yep. Okay,
perfect. And Mark and Ken, thank you so much for inviting me to present as part of this wonderful workshop. Really enjoyed yesterday's presentations as well. So as Dan said, I will be talking about genomics-based clinical informatics resources to support precision oncology and evidence curation. So these are my quick disclosures. Just let you look at it for 1 second. So in the next few minutes, I wanted to share with you all 2 stories. The first story is around a virtual tumor board. And here, the focus is really on facilitators and barriers to deploying genomics-based clinical informatics resources in the clinic in cancer care. And then the second story I wanted to talk to you is much more sort of on the informatics research side, is in terms of identifying research needs and how do we sort of deploy genomics-based clinical decision support in the clinic, and what research needs to happen in order to make those clinical decision support systems work really well. So the first case study is about this 52-year-old male. He was diagnosed with stage 4 pancreatic adenocarcinoma. He was being treated at the Lombardi Comprehensive Cancer Center here at Georgetown University. He was progressing very aggressively. His disease was progressing very aggressively after standard chemotherapy, which was, typically gemcitabine, nab-paclitaxel, which is given to patients with pancreatic adenocarcinoma. Then the team conducted a multi-panel genomic testing, and sort of, thank you for telling me it's cancer. It came up with KRAS and P53, etc. But a deeper clinical informatic analysis revealed a mutation in a VEGF receptor, which resulted in selection of a clinical trial that really extended this gentleman's life by a matter of few months. So, people who know pancreatic adenocarcinomas know that the progression-free survival on average is about 12 months, and any improvement to that is considered a big benefit. So in this case, the patient had a VEGF receptor mutation, and the team identified a ligand-independent constitutive phosphorylation, which led us to select a TKI, a very specific tyrosine kinase inhibitor that targets that particular pathway. And he was able to be enrolled onto an NIH-sponsored sorafenib trial. And this was an email that our patient coordinators received from him. And he was almost ending up in hospice, but without this genomic analysis and the clinical informatics behind it, he would not have benefited from this clinical trial. So the question was, how do we scale this to 300 hospitals across the country and over 1,500 patients? These were all patients that were part of the Know Your Tumor program within the Pancreatic Cancer Advocacy Network. It's called the PanCan network, and they work with centers across the country. And the question for the clinical informatics team was, how do we scale this experience that I just showed you to all these patients who come to PanCan? And the solution that the team developed was an artificial intelligence-based virtual molecular tumor board. And you can, I have these references here, right here below on my slides. You can read about them in more detail. But very briefly, the AI-based virtual tumor board involved development of a virtual knowledge base and collection of molecular profiling data from patients, as well as clinical information, their clinical history, other comorbidities, and demographic data, et cetera, from the EHR, combining that information and presenting it to a medical review panel. And the virtual tumor board facilitated the workflow of this medical review panel, which then finally developed these individualized reports for each patient. And then the patients were later on followed for real world outcome analysis over a period of 6 months. And that data got incorporated back into the knowledge base to help other patients who came into the PanCan Know Your Tumor program. So here is very quickly the information engineering that was used for the Know Your Tumor program. So everything that you see in yellow is publicly available information. So this is your clinicaltrials.gov or published literature, guidelines from NCCN or the FDA, other published data about somatic variants, and the evidence associated with associating those variants to various targeted therapies and so on. And what you see in blue is the information that was collected from the patient. So the molecular profile and the demographic data, et cetera. And this therapeutic engine integrated the public information with the patient-specific information and analyzed a number of rules for treatment implications and provided the recommendations to the medical review panel, which was then reviewed by humans, the doctors, and entered into this particular report. So now the group has started to think about, how could we incorporate this concept of virtual tumor board in every medical center? I mean, we know that cancer care, majority of cancer care actually occurs in community hospitals, and not all of them go to academic tertiary care facilities. Very rarely people go to academic medical centers. And so the question is, how can we leverage the expertise that's present in these academic medical centers or other tertiary care facilities in specific clinical genomics areas and provide that precision care to patients who go to community hospitals. And so this is a workflow, which was a concept that we wrote up in JCO-CCI last year, but slowly components of this are beginning to be implemented within community centers. So for example, I'm part of the MedStar Health clinical system, which is 10 hospitals and 240 prompt care facilities in the greater Washington, D.C. area. We are one academic medical center, which is the Georgetown University Hospital, which you see behind me. That's the Healy Hall on the Georgetown campus, but rest of the hospitals are all mostly community-based in Washington, D.C. and in Maryland. And the question is, how do we take what we've learned at Georgetown and implement it writ large at these community hospitals? And here is where our work, which is funded by NHGRI, comes into play. So the ClinGen and GA4GH VIC communities, VIC stands for Variant Interpretation Cancer Consortium. These are communities of experts who come together to create these evidence items for the somatic variants and associating them to various therapies, and also assigning these variants into different tiers. This is a tier 1 variant. So there is a direct association to therapies, there a tier 2 variant or a tier 3 variant, and so on. And then that information can then be made available to the treating clinician who's taking care of the patient at this community hospital. So what are some of the barriers? So this project has been ongoing for almost 4 years now. It started out with Know Your Tumor program and really expanding out to other centers, both in the United States and across the world. And some of the major bottlenecks that we see are, there's a lot of manual curation. So as you can imagine in the information engineering slide that I showed you, there's a ton of manual curation and data integration. So we need armies of people who are reading literature, who are, pulling data from databases and integrating them into this knowledge base. And there's a clear need for consensus and variant interpretation. And I don't think this is news to anyone here. I mean, there is all kinds of databases for variant interpretation. People have specific expertise in different domain areas. So there's a clear need for evidence adjudication. And the machine can only go so far. So I kind of say that, it can get us to 80% of the way, but, the real human input is needed when clinical interpretation is required to treat a patient. Too many knowledge bases. This is extremely onerous for clinicians and phase 1 clinical trialists who have to adjudicate all this information. And it's very onerous on them to be mining these multiple knowledge bases when they have to develop a treatment plan or think about which clinical trial might be best suited for this patient. Lack of resources for curators who are creating these one-off solutions. We have a number of curators that are part of the ClinGen consortium, for example, who are manually curating evidence for pathogenic variants and assigning the different evidence tiers. And there's a lack of easy-to-use, user-friendly interfaces for them to do their job. And that is definitely a rate-limiting factor in constructing this knowledge base to support clinical decision-making. Need for more uptake to clinical researchers. Eric yesterday talked about the need for training and diversification in training. And clearly training is needed. Sorry?
This is your 1 minute warning.
Oh, 1 minute. Okay, thank you. Need to uptake within the clinical researchers. So it takes an army to actually get this work done. So this is face-to-face meetings at our ClinGen consortia meetings where we come together and really create these knowledge bases for the evidence that I talked about. So here is our first publication. We call it MVLD, minimal variant level data. This is a standardized format for representing evidence for somatic variants, and there was a very big uptake of this as soon as it got published. So I think I misunderstood the amount of time that was allocated. So I think what I'm going to do is skip the second story and just show you one picture. And then we will stop there. So in terms of what is the research focus? So where do we really need to put our focus to really enable this clinical decision making? And here is an example of a proof of concept that we're developing at Georgetown. The font size is small, but I'll just sort of high level describe this, is that this is an NLP pipeline, a natural language processing pipeline that uses this concept of learning from less data, because we don't have millions of records to learn from in most cases. So when you are looking at one particular disease, for example, pancreatic adenocarcinomas, very specifically VEGF receptor, you don't have that many articles to learn from. So the question is, can we learn from less data? We all talk about learning from big data, but can we learn from less data? And there are some very interesting research that's coming out in learning from less data to really enhance clinical decision making. So you can learn more about that at this website that I've shown here. So just wanted to say thank you to all the folks that contributed to this, and this is the team that worked on the MACE2K as well as the Virtual Tumor Board project. Thank you very much.
Fantastic talk. I'm always in awe of your work. You have several questions. Mark actually has a 3-part question. Mark, do you want to ask this directly? Would that be easier?
Well, actually, Suba addressed it right after I hit submit in the next slide. But I think the question that remains would be, you referenced the manual processes both in the patient data and in the literature review. And I think you have referenced a little bit about learning from less data, but what would you highlight as being the key areas for research to try and reduce reliance on these manual processes?
Yeah, that's a great question, Mark. So some of the things that we are looking into is, the concept of semantic abstraction, right? So, there may be a lot of associations that are present in these knowledge bases or in literature, but we need common data models that can help us understand that two sentences may be talking about the same thing. So for example, the association of a VEGF receptor with a pancreatic adenocarcinoma may be stated somewhat differently in one knowledge base versus another. So are there common frameworks for the semantic abstraction that we could learn from, so that so that when we feed this into the machine understands that two different sentences that talk about the exact same association in two different ways, in fact, mean the same thing. So I think, how do we sort of standardize this interpretation of variance is one big area that we are focused on, and a number of people, and I see colleagues from GA4GH participating here as well. We're all kind of thinking about how do we standardize this framework for evidence adjudication and standardization of this evidence.
Fantastic. And there's another question. Does the VMTB have the opportunity to capture outcomes in response to the recommendations made, such that the community learns and optimizes recommendations?
Yeah, that's a great question. So one of the things that the Know Your Tumor program within PanCan is attempting to do is, after the reports are delivered for a period of 6 months, they would collect data back from either the care team or the families. So essentially there is a little app that was used for the patients to enter data. One thing to keep in mind is, patients with advanced metastatic cancers are very sick patients. So it's very hard to collect outcomes from these patients directly. And so the app was essentially made available either to the care team or their families for them to enter information about these patients. And that gets incorporated back into the knowledge base to support. I mean, we had about 30 to 40% completion rate for the forms or the surveys that were sent out. So we don't have outcomes on all patients, but for some patients, definitely. And whatever outcomes came in, that goes back into the learning process, into the machine learning process for the future iteration of the knowledge base.
Fantastic. I think we'll take just one more question. How parallel are the data models between germline and somatic variant curation?
Yeah, so that again is a fantastic question. So we are doing a lot of this work as part of the somatic, we call it CDWG within ClinGen, the Clinical Domain Working Group. And majority of the work within NHGRI supported ClinGen is all germline focused. So what we're doing is we're taking the frameworks, the ACMG frameworks and the actionability frameworks that have been developed over the years for germline, and making modifications to suit the somatic interpretation. It doesn't fit, there's no one-to-one mapping, of course, but there's a lot to be learned from the germline interpretation for us to make the somatic interpretation. And why is this important? This is important because in the cancer world, we're not dealing just with the somatic mutations. We're also dealing with germline, and there's constant germline filtering that needs to happen even when you're looking at tumor biopsies. So if the frameworks are matching, it's easier for us to not only look at the somatic variants, but also in the context of the germline background, which is then represented in the same framework.
Fantastic. Fantastic talk. Great discussion. Looking forward to the broader discussion after our last speaker. So with that, I think, Dan, we should transition and introduce our last speaker, and then we'll open it up for a broad discussion with everybody.
Okay, so our last speaker is Neffy Walton, who is going to speak to us about the integration of genomic data into electronic health records, current state and future directions. Hold on a second. My electronics are screwing me up, so I can't. Sorry. Give me 1 second. Well, I want to introduce Neffy, but I can't find the, there we are. I'm an idiot. So Neffy is currently, he had led several genomics and informatics initiatives at Geisinger and Intermountain, and is going to talk to us about heredogene with plans to sequence 500,000 patients.
All right, so my talk is actually going to be more about the integration of genomic data into the health record based on work I've done at Geisinger and now with Intermountain and Heredogene. As my disclosures, I do serve as Associate Medical Director of Intermountain's Precision Genomics Sequencing Laboratory that does offer commercial sequencing services. I do not have any affiliation with or financial interest in any EHR vendors. In fact, there are times when they would pay me not to speak. Mark will attest to that. Opinions and viewpoints are my own and do not represent those of any EHR vendor. So anything that I talk about from about an EHR vendor, that's my perspective and not necessarily the stance of the EHR vendor. As a historical background, I started at an institution that used Allscripts, moved to an institution that used Epic and implemented genomics, and I've now moved to an institution that uses Cerner and I'm implementing genomics again. The vast majority of genetic data remains in PDFs today. So at Geisinger, we completed a genomic data integration pilot. We imported about 1,200 patients' genetics data, including tier 1 variants and pharmacogenomic variants. And we built clinical decision support and patient provider facing information for all of those variants. We learned a lot in the process. There are a number of challenges that we faced and that I think that more research is needed around it. I'm going to discuss those quickly. I won't be able to go over all of them, so I'll put the reference to the paper at the end because there's a lot to go over. So one of the first challenges we hit was the difference in the needs of clinical geneticists versus primary care providers. As a clinical geneticist, myself, I was looking for a lot of things that weren't there in our initial implementation, but the implementation was suited pretty well for population genomics, which was suited well for eMERGE and the MyCode program. What we need to do in this area is define separate but compatible workflows for different genomics use cases. So if we look at the EHR vendors and their different approaches they take, it's really interesting. So Epic has a system that's in production that is really geared towards population health. Cerner has now developed a system that's in the alpha phase that I've seen that is built by clinical genetics and therefore is very geared towards clinical geneticists. Those are very different workflows, and I think it's important that both of them are developing out in different ways and actually could learn a lot from each other. Allscripts took a different approach initially, and their product has matured a bit since I've looked at it last, but they were really looking at proactive testing. So they would look for patients that could use genetic testing and suggest it at times was one of their first approaches. So it's really interesting. They took 3 different approaches, which are all valuable and frankly, all important. And if I could say one thing that was really needed in this area, it would be implementation science and looking at different ways to implement genomics for different providers, including primary care, clinical genetics, and other subspecialists. The major challenge that I think the EHR companies are looking at that think is a major barrier is the interface between the laboratory and the electronic health record. Right now, most systems require that you manually enter genetic tests reports into the system, and that's not a viable solution. Someone on my team suggested hiring genetic secretaries since we have genetic counselors and genetic counseling assistants. This is a bad idea. I think he was joking. What we really need to do is work on interfaces and standards. So again, here we have 3 different vendors and we have 2 different standards in this case. Epic uses HL7 v25, which is kind of a legacy standard. Cerner is using FHIR and Allscripts is also using FHIR. And one of the arguments is that it doesn't matter what you use because laboratories don't support either. And actually laboratories were saying the same thing. It doesn't matter what we use because EHR vendors don't use either. I think this is starting to change and I'm hoping that everybody moves towards FHIR. And that will be overcome a major barrier. But even if we use those standards, there's another thing that's a major barrier to implementing this in clinical practice. And that is the standard definition of genetic phenotypes. So when you get a genetic test report, you get all kinds of names for what might come across as your genetic phenotype. So different naming conventions, for example, if you have a BRCA1 result or a BRCA2 result, they could be named different ways. And so that comes across as a genetic phenotype. What we need to look at genetic phenotypes as is a point of decision support. So you're saying, based on this genetic phenotype, these are the decision support you're going to offer. This is the information you're going to deliver to the patient. Now that can be different by the gene or the variant. And so having some type of ontology or model built for a standard for genetic phenotypes is really important. So we built this out for one laboratory, but most institutions actually have, at least 5 or 6 different laboratories that are returning genetic results that are all using different genetic phenotypes. So we need to rethink this area and what the point of decision support is for different diseases. Challenge 3 is the maintenance of clinical information and clinical decision support. So this is a huge area of need. There's not a lot of publicly available resources. And one of the big problems is that currently maintenance of these types of system requires technical skills. You actually have to bring in the EHR team to help develop, to maintain these resources instead of people who are experts in genetics or experts in pharmacogenomics. And as many of you know, those resources from the EHR development team are very rare and hard to get. So one of the ways that we can help this is to build open APIs where you can have people build external applications where it's easier for people to maintain and curate those things. Challenge 4 is classification and reclassification of variants. So this is a huge problem from a number of different perspectives. So one of the major challenges here is that there's a lack of communication between the lab and the clinician. So the reason why this is important is often a laboratory will give a report that says this is a variant of unknown significance. Well, the clinician, in light of having the patient in front of them, can often make a determination that yes, this isn't actually a pathogenic variant. Well, what happens then is they might reclassify it and say the patient has a disorder. That information never goes back to the laboratory, and it's actually possible that they could have 2 discordant classifications in ClinVar for the exact same patient. Likewise, there's a potential for discordant results between a PDF and structured data in the electronic health record. As a PDF comes from the lab and then it gets reclassified by someone, so a clinician, and put into the HR, you'd have discordant results which can be confusing. And further, that gets complicated as you have patients that have the same variant, and you might reclassify the variant based on a patient presentation, and you'll have 2 or 3 other patients in the system that will have a different classification, which opens you up to all kinds of challenges around liability and treating patients in different ways. Another area that I wanted to touch on was the moving beyond the initial implementation and looking at the real-time use of genetic data. So this is something that we have a grant submitted around, and also something I'm working on as an initiative at Intermountain, where I'm trying to really interface the laboratory into the clinical space where I have that opportunity. And some of the challenges that we face in this, one that has been touched on before, excuse me, is accurately capturing phenotype data in a structured manner. Because to be able to do this, you have to have a lot of computable data, and if it's not structured, it's not computable. There are also a lot of challenges of using genomic data with machine learning and artificial intelligence. This is something, as chair of the Genomics and Translational Bioinformatics group at AMIA, that I'm working on a paper to discuss this. Genomic data actually introduces, there's a lot of challenges. First of all, you have this huge data space that you're working in. And so machine learning, I mean, in ways that it's good at using where there's a lot of data, but the problem is we don't have a lot of knowledge surrounding that data. Another thing that was touched on earlier is the potential racial and ethnic biases. I mean, I think genetic data is the most probably dangerous thing in that area, because there are a lot of genetic similarities between people in different racial ethnic groups, and you'll actually be making decisions based on that genetic data, and that has an inherent bias, and to be careful that that is used in an equitable and ethical way. The other is the regulatory nightmare surrounding the use of entire sequence. So, we have all of this huge trough of information about a patient that what drugs they're going to react to, we can use real-time genomics in their diagnosis. We can use it to essentially personalize care. But the problem with that is that everything right now is based on a clear report that comes from a laboratory. And in order to use that other data, you have to go back through that process. So we need to look at some ways to streamline the use of genetic data outside of that clinical report. And I actually went through that faster than I thought I would. I don't know how much time I have left. I kept going slightly over when I timed myself. I must have beat my record. So anyway, I have a reference at the bottom about the paper we did regarding our implementation at Geisinger, and I'll open it up to questions.
We have a question regarding the genetic phenotypes. Is it possible to define a genetic phenotype from a variant alone, or does this require information about the patient's phenotype? If the latter, do labs have enough information to make a call?
No, I mean, I think that we need to start. So when we look at genetic phenotype, this is the point I'm trying to make, is that this is a point of clinical decision support. So this is a point where you're saying, based on this genetic phenotype, we're going to care for this patient differently, right? And so for the most part, a patient with a BRCA1 variant, they're going to be treated pretty similarly. Now, for some disorders, there are modifier variants that will modify their risk. So they might not require certain types of surveillance, or they might require more surveillance, depending on what that risk is. And so it's something that needs to be really well defined and curated, in my opinion, because there's for any given disease, there's thousands of potential variants. And so we need to say, through research, this variant confers a different risk, and we need to be able to have some sort of database or ontology to go to say, okay, this needs to be a separate genetic phenotype. Does that make sense?
Does to me. Last question for you, and then I think we'll open up the questions to all the panelists after this one. What would you advise as the most valuable next steps to address the regulatory nightmare you referenced?
I think to tease out pathways for use of clinical genetic data and potentially even a risk analysis, I guess. And maybe just identify, here's how we'd like to use it, and here are the barriers. I haven't thought of good ways around the CLIA report yet. I mean, if you open it up and you have no regulation, there's a lot of misuse, and I think that's why FDA has taken on pharmacogenomics. But then I would argue, if the government is going to regulate it, they need to provide some really clear guidelines and even offer that database that you can access to say, this is what you can and cannot report, and the extent to which you can go. Because even in pharmacogenomics, I used to think that's an area we've got really well defined. We've got good clinical decision support guidelines. We've got good information. But even in that area, now that I've become, it's the associate medical director of the laboratory, and we're doing pharmacogenomic testing, you realize it's still not clear. And even FDA regulation and how all that's going, that's still not clear. There's a lot of gray area, and there's a lot of things that need to be clear there.
So I think at this point in time, we will open this up for all the panelists to answer questions and to partake. I want to go back to one of the questions that was asked earlier. Is there an initiative similar to CPIC in the cancer world? Is anyone working on updatable peer-reviewed guidelines that match somatic cancer variation with anti-cancer drugs? And that's for all panelists. That's for all three of you.
Yeah, and I can start, and maybe others can join. So there are a number of groups that are looking at sort of associating cancer variants to various therapies, right? So the My Cancer Genome is one at Vanderbilt. There are a number of such organizational databases. There's OncoKB, which is led out of Cornell. And the VIC organization that I mentioned briefly, the Information for Cancer Consortium, which is actually a driver project within GA4GH, the Genomic Alliance for Genomics Research, the Global Alliance for Genomics Research, is a driver project where a number of these databases are actually being integrated. So the VIC group is creating standardized data models and APIs that connect across these databases. the barrier that I mentioned is that each organization that's doing this curation kind of focuses on their areas of expertise, right? So in our case at Lombardi, we primarily focus on breast and GI cancers. So all of the curation that we do is pretty much focused on those. And you kind of see those silos. And hopefully the APIs and the standardized models that Vic is developing can interconnect these, and we have a good representation across these different domain areas.
And I'm curious, yesterday we heard a lot about, issues with algorithms lacking the ability to handle diversity and biases and so forth. So for your project, Know Your Tumor, what's the distribution of the different, ethnicities represented within that algorithm and that AI machine learning platform that you mentioned? And what did you guys do to ensure that those type of biases were not inherent within this platform that you've established?
Yeah, that's a great question. I have to go back and look at the numbers, but I think it was sort of just the national representation of pancreatic cancers in the United States. Those were the percentages. The beauty of this study, because it was all comers, so so there wasn't a design that kind of made sure that there was a balance across different ethnicities. So it's all comers, patients with advanced metastatic cancers who needed a precision-based approach for therapy, but there was a good distribution as far as I recall. I have to go back to see the exact numbers. But I think after the fact, after the information was collected, we did some subset analysis to see that were there more actionable markers that benefited patients with certain ethnicities and race groups, etc., which now is being fed back into the knowledge base to support other people who are coming in through the KYT study.
Fantastic. We've had several questions come in, and I'm going to just try and summarize in some of the discussion points. I hear a common thread here, and I'm going to call it a common data model thread, although we're not using that word often at all during this session. But to me, when I think about, the first speaker, Teresa, and your work and some of the issues that we're hearing from Nephi, I mean, to me, this sounds like a common data model, and we're lacking common data models. We're lacking the ability to structure our data in a format that can be shared across institutions or within an institution to drive research. In order to drive research, we need to get the data out of the EHR into a standardized format. So I'm just wondering, across all the, the speakers in this session, again, we didn't talk much about common data models, but what's the role of common data models? How are we going to solve this problem of creating this sort of structured system and to be able to handle this type of work to drive research?
So I think that we do need, and there are a lot of data models and standards out there, and some of them do a pretty good job, and I think a lot of it is going to be people working together. And part of the challenge is this has only recently really entered the clinical space. It was not long ago that everybody had only a PDF in terms of genetic data. So it's only recently come in structured format within the HR. So it's young, and I think that it will mature. I think the biggest challenge is to get everybody to adopt the same data model and agree on what a genetic phenotype is. I think that's a really challenging concept for people to understand and get a handle on. But it really needs to be, I mean, the reason that you would have a different genetic phenotype is because you're going to make it, in my opinion, is you're going to make a difference in care. And now, for research, that's different, because I mean, one of the things we also need to look at is, where we need to find out if there's differences in care, right? So we need to start looking at people with different variants or variants in certain domains, variants in certain genes, to see if there are different ways that we should care for them. That's one of the reasons I'm a big proponent of more aggressive genetic testing. A lot of people say, well, we won't change care. Well, we'll never know if we're going to change care unless we start looking at these cohorts of patients with these changes to see if it will. So I don't know that there's a great answer to that. I think that things are improving over time. I think the standards through HL7 have changed dramatically, and they're much better than they were even a short time ago. So I think things are getting better.
And to build on Nephi's comments, I did want to clarify that once USCDI is implemented across health IT systems, researchers will at least be able to bank on the data classes and elements required under USCDI to be standardized, right? Those are underpinned by controlled vocabularies, and those are specified in the standard. And so as it evolves, then there's an opportunity for it to either add layers of specificity or add data classes or data elements that are needed broadly for clinical care and that would also benefit research.
Yeah, and with that said, I mean, that part of it, there's a difference between a data model too and the knowledge or content or, that we put into it. And that's an area that I think, you know, needs a lot of development too, to have sort of public domain resources for agreed upon knowledge or and I know it's, you know, this is an evolving space, but somewhere there needs to be kind of a consensus, especially if there's going to be attempts to regulate it.
I think I would just quickly to add to what Nefi and Teresa said. While it's important to have these standardized data models, it's also important to have apps or applications that use them, right? I mean, that's how the end users and the clinical decision support actually can happen. So that's one point. And the second one is, I think Nefi is trying to say this, is that the data models need to serve a lot of the data. I mean, I think the holy grail for machine learning is really labeled data. And, you know, unless these data models serve a lot of information or data, they end up not being used. So those two would be critical for the successful data model to kind of emerge as the one that people uptake and use.
The one to rule them all. We have a question actually. Oh, I'm sorry, Mark. We have a hand up and an attendee, but do you want to go first, Mark?
Yeah, the challenge, of course, with this discussion is always to try and contextualize it for the purpose of the meeting, which is we all recognize the need for data models, standards, infrastructure to support both clinical care and research in this space. But the question for this meeting, as I see it, is how can this be framed into a research question or a research agenda? In other words, how can we fund research to move us forward, and what would be the metrics? So I'd like to hear some comments from the speakers on that.
I think, Mark, there's a lot of implementation science here, because I think that's how you discover and find out what's needed. I think my view of what we needed and how to move forward was a lot different before we started the implementation at Geisinger and than it was afterwards. And the only way that you find out what's needed is to actually do it, to do pilots, to do research studies. And so I think there needs to be a big focus on implementation science so that we can discover what's needed and run into the challenges. Otherwise, we're never going to see them.
So putting on my human factors engineering hat, even a step before that is really identifying the user base and their needs, right? So when you start to pilot something, you have something, a tool, a system, whatever it is, an artifact to pilot test. But an HGRI needs to think about what the Institute sees as its stakeholder group, who it's trying to serve, whether it's the research community, the genomic research community specifically, or the participants, or broader than that, and start thinking about identifying the needs of those communities to inform an agenda around developing and pilot testing some of these tools. And then related to that, we've talked about standards and common data models. Part of the issue we ran into during Think for Genes were some issues on lack of semantic consistency or clarity. So understanding where there's consensus and where there isn't, and where additional research might be needed or work might be needed to arrive at some of that consensus to help inform next iteration of standards.
I think, Mark, if I have to bet on something in the clinical informatics realm for genomics, I would say it's explainable AI. Every time we build these machine learning models or artificial intelligence models and present it to clinicians or end users, the first question is, what are the features that led to this precision or this recall or this F1 score or this sensitivity or specificity? I think the better we as a community are able to explain the features that are actually resulting in certain predictions, I think that there is critical research needed in that space. How do we create these explainable AIs, which are not black box deep learning models that end users can understand and use within their workflows.
No, I'll go. I agree with, I totally agree with that. And I think the better that AI becomes, the more dangerous it becomes, because we start relying on it more without using our own brains. And we have to remember that AI is based on what it's seen. And when you run into situations that the machine hasn't seen, which happens often in medicine, and you trust it, then you run into problems.
Great. I'd really like to transition a little bit because we have an attendee who's had their hand up for quite some time that would like to ask a question. So Suzanne, thank you for being so patient. If you'd like to take yourself off of mute, because I can't actually see your question, I just see that you have your hand raised. Would you like to ask your question now?
Yes, thanks. I'm sorry, I'm just joining you today, and my question's in a little bit different area, but I think it was opened up a bit by some of Teresa's comments for the 4th stage of Sync4Genes, and also a comment that Dan made as well. And I'm particularly interested in more the consumer facing angles. And particularly in thinking about returning reports to patients, research participants, and caregivers, and certainly individual institutions such Geisinger do a good job on this, both in eMERGE and in all of us. There are experts pulling together to create, I would say, attempt to create culturally congruent, health literate messages, as well as visualizations around related to risks and other aspects of these kind of results. But as a kind of a research agenda, I think we really need to be moving more towards a computable, like, visualization and message toolbox that would allow a variety of sites to be able to use a component, specific infographic components, and to build some tailored messages. So I just want, it's fine if there's any reactions to that kind of notion and where it fits within a clinical informatics research agenda.
I would just add one thing to that as well for the panelists to think about is, should we have some of our community members attend these types of forums and have them represented and do presentations on the consumer end, so we make sure that their viewpoints are incorporated in our research agendas. So with that, I'll open up Suzanne's question and those follow-up comments to the panelists.
So this is an area of a lot of interest to me. So we deliver results to patients through the Heredigene project, and then I also was formerly from Geisinger, where we were delivering it through the patient portal, and that is so that's one of the major problems. If you're an institution and you're trying to implement genomics, one of the first things they're going to ask is who's going to own this and who's going to curate this. And most institutions don't have the resources or the people who can do that. And especially as you expand it to, hundreds or thousands of genes. And that's where, I had the point in my slide about that there are not a lot of public resources. And I think it would be great to have databases that had that information for patients and, frankly, providers. Providers are also not real well informed. There's a lot of primary care providers that manage some of these patients, particularly if they're not close to one of the main medical centers. And so having some type of resource built that stores and provides that information for hospitals would be great. And I think that'd be a great area to focus on.
I mean, the cancer world has been doing this for many years, right? So the patient advocates are constantly part of many research groups, if not on a regular basis, at least they are quarterly meetings, et cetera, where we meet with patient advocates. One thing that the Know Your Tumor program did was, while there was the 65-page clinical genomic report that was delivered to physicians, there was a 3-page summary that was presented to patients as well. sort of a, a 6th grade reading level layperson language was incorporated. I'm seeing some new innovation that's coming out in the virtual tumor board space where, now some of these apps allow for patients to be part of the discussion, at least for some portion of it. And I think there are pros and cons to that. But some apps are starting to think about, if my case is being discussed, should the patient be part of that discussion when the surgeon and the oncologist and the multidisciplinary team is talking about. I think there are lots of other legal and liability issues that need to be considered, but I think people are definitely moving in the direction of incorporating the patient more in the CDS.
And so then based on AI, go ahead.
So
Go ahead.
sorry, Erin. Yeah, so we actually have patient representatives and the technical expert panel for Sync4Gene Phase 4 currently, and we sought out individuals who truly represented patient views, and particularly within the genomic space, given the nature of the project. And it's something that we're doing more systematically across our work. We have other projects that are very much patient-focused, developing platforms for apps and tools to share information with them, and those are going out into communities, and actually pretty diverse communities, to ensure that we build tools that work for a lot of people and not just a select few. Sue, to your comment, I think there's definitely an opportunity for an informatics research agenda specific to how to build and scale similar tools for patients and caregivers. I can't speak to, whether a toolbox or something similar is the right approach, but I think it's a question worth asking.
So as we build more clinical decision support tools, particularly proactive ones, how do we develop a responsibility model to decide who is responsible for ensuring information gets to patients?
So, I mean, I'm not sure I completely understand that question. So I think that one of the big needs now is having, especially if there's regulation, like I mentioned, having that information out there, having somebody curate this information so that even smaller hospitals and primary care providers can have access to it. And maybe that becomes a commercial venue, but it would be nice to have some resource out there. I know, at Geisinger and at Intermountain, we're looking at several different methods to deliver that to patients. And, we have the patient portal, not everybody accesses that. We mail documents, we have a chatbot platform, text. I mean, and part of that is, you have to work through your regulatory processes as well.
Yeah. So, I mean, patients have information now, right? They're getting their being sequenced already. They're going to the private sector. So I think the issue is building tools to help them interpret information within the context of clinical care, and ensuring the healthcare system shares results in a timely way with the necessary tools to help an interpretation and have a conversation with your clinician.
Great. Is there an admin on here that could enable Heidi's audio? She's the one who asked this question, and she'd like to clarify it. She's coming through the chat box and the Q&A. I don't know how to enable Heidi REHM's audio. Is there somebody on the technical side that can help me do that?
Yes, I just enabled her audio.
Thank you, Pam. You're the best.
You're welcome.
Sorry, can you hear me?
Yes, we can, Heidi.
Sorry, the question was more around, we have this issue when an actual patient's report gets updated from the issuing lab, a very direct feed that needs to go to that patient, but the ordering physician has retired, left, or isn't caring for the patient. How do we think about a responsibility model across the EHR so that patients will ultimately get the information they need, in these settings where we're thinking about proactive, preventive, notifications that have to go to a very specific patient from a very specific source. I'm not talking about vetting knowledge sources. I'm just talking about this direct feed.
Right. I think that's a major challenge, and that was addressed by the ACMG attorneys. I'm sure you were at the meeting. Where does your where does a clinician's responsibility end in terms of caring for a patient, returning results if something needs to be updated? And if the patient falls outside of that patient physician's care, what happens then? I know there's been some talk about having, from, I won't name the vendor, about having that direct connection with the patient so that they could deliver any updates to their genetic test report. I think that, I don't know that I have a good answer for that through healthcare system, but there definitely should be a way to notify patients, and perhaps what we need to look at is other areas in the HR where that's happening, or something similar is happening, and look at some models that exist. Off the top of my head, I do not have a good answer for that one. I don't know if anybody else does.
I mean, I've always thought about it in the sense that, if an alert goes out and nobody responds to it in the HR, that there is some time period before which the patient gets pinged and says, you should contact your physician. There's information that might be of relevant to you, something generic, but triggers the patient to ultimately have to come back in. It's always the way I've thought about it, but I feel like there needs to be some regular, governance model to how we think about these types of interactions. Right,
and the challenge with even that is that a lot of the patients, for example, through at Geisinger, they're not, the regular healthcare doesn't happen through Geisinger. They might have come for to see a specialist, but they actually have a primary care somewhere else on a different CHR that manages that. And I think, ultimately it would be nice to have a patient have a portal their own data and have some standards there so that whatever lab could send updates to the patient about their genetic data that came up. But then again, you have all the regulatory concerns about, and then the concerns about follow-up and whether they'll interpret it right. There's just a lot of challenges around that.
Yep, I agree. There are challenges, and I think we all need to
think through them. The point being, this is an area that needs a lot of research.
Yes, that was my ultimate point.
And we have somebody chatting who would like to, based on Heidi's point, I'd like to take that one step further. What if a result in the exome is relevant for disease that's different from the clinical area for which the original test was ordered? We need a model where the result goes to a provider that can implement care for that patient.
Yeah, so this is something that I am working on at Intermountain. And again, this is not entirely straightforward, I will say, because there's a lot of information and some of it won't be relevant. So we return all of these conditions to the ACMG reportable conditions and a few others, but there will be other information that becomes valuable over time. And there will be information that is valuable when the patient exhibits a certain phenotype that might not be valuable without that phenotype, such as epilepsy is a good example. So having something work that way requires a very tight collaboration between the laboratory and the clinical space. Again, that's something I'm working on, but it's not at all straightforward, is all I'll say. So that is an area also where a lot of research is needed.
I'd like to then transition to Ben. Ben's had a couple questions coming in and out. Ben, would you like to unmute yourself at this time and ask your questions directly?
Sure, can you hear me?
Yes, we can.
Great, thanks so much, and thanks for the chat back and forth, Mark. So I guess my question is, in artificial intelligence and machine learning, it's very exciting to see what can be done from data sets that are where the answers are known, but also with zero-shot learning, with applying these, as we discussed, to things that the computer could infer that it hasn't already seen, whether it has to do with natural language processing, machine learning. There's a lot of interesting stuff on paper that this could work pretty well, but in reality, Mark, as you mentioned, do you see this as something that could be tried, should be tried, is too fraught with issues? I was just wondering, Suba, especially in the work that you're doing with cancer genetics, if you see this, are you already doing this, or is this too challenging for lots of reasons? And thanks, and sorry I kind of articulated this pretty badly. I know there's a lot to unpack here.
Yeah, no, I completely agree, and I think it's a great point. I mean, I think we kind of need both approaches, right? One is sort of the supervised learning, where we know the classes and we are predicting the classes that are already known. And then the second category where you're actually letting the data tell you what the associations might be. And I think large companies like Google and Microsoft, et cetera, are building these knowledge graphs just as an exploratory tool, just based on the searches that people are doing and the other data that they're collecting. So I think we need both of those approaches. For the second one, we of course need very large amounts of data for us to really infer those relationships and those networks. And at least in my world, we start out with these petabytes and exabytes of data, but by the time you arrive at a data set that you can work with in your Python program, you have, I don't know, less than 500 cases, and I'll be happy if I got very good labeled data sets with 500 cases. So yeah, I think we definitely need both. And I would even hedge my bet and say that the second area, which is the unsupervised and the exploration and more discovery, needs more research, right? Because we definitely need to explore that and see what the data can tell us. Maybe those are not the associations we're looking for right off the bat.
These are not the droids we're looking for either. I typed a response to Ben, but wanted to kind of weigh in that I think that the place where I would be very excited about some research is actually at the interface of humans and AI, because a lot of the studies that have come out have, the headline says AI outperforms physicians at X task or Y task or Z task, which is true, but the thing that's missed is that in most of those studies, when you combine the AI with the physician, it outperforms both the physician and the AI. And so to me, what that's saying is that the AI approach and what we do in human cognition are actually complementary. And I think that that's been very under-researched and is a nice opportunity. And I mentioned in the response that Nephi and I had worked on a project where we were looking at a diagnostic decision support tool for rare neurogenetic disease, where you would enter information, the system would prioritize a differential diagnosis, which is nothing new. But the other thing it would do, it would say, if I had this information, this would have the biggest impact on this differential diagnosis. If you could answer this question for me, or these 5 questions for me, I could give you a very informed answer, but right now that data is missing. And so anticipating what the data needs are that could then prompt a clinician to say, oh, I should either order that test or I should add this information because I didn't think it was important, illustrates how that interface could potentially be very productive.
I totally agree. And I just want to add to that, the important thing there too is that with the artificial intelligence, that the provider, the clinician, has some information about how that decision was reached and why the machine is making that decision. Otherwise, they're not going to blindly change care if they see something that flies in the face of what the machine is telling them. So they need to be able to have, and I know it's complicated and often black box, but there needs to be some understanding there. And that, again, I think that someone had mentioned that earlier, we have to be able to explain these models and why they do what they do.
So I think we're pretty much at time for this discussion. I want to thank the presenters for the session for their very interesting and provocative presentations. Thank you to our co-moderators, Drs. Krauge and Roden.
Mostly Dr. Krauge.
Huh?
Mostly Dr. Krauge.
Yes, well, I'm just trying to be polite, Dan. Those of us who work with you on a day-to-day basis, we all know, but we'll try and maintain the illusion for the rest of the audience. In any case, we're going to now take a 10-minute break for people to be able to get up and stretch. So we'll be back at 1:35 Eastern time with session 5. So thank you very much, and get the blood circulating and don't throw an embolus.
Get started. So thank you, everyone. So we'll start our session 5, which is actually genomics in a fragmented healthcare environment. And our co-moderators for this session is Dr. Freemuth and Dr. Jeffrey Ginsberg. Dr. Freemuth is an assistant professor at biomedical informatics at the Mayo Clinic Center for Individualized Medicine. He's also the co-chair of several work groups, including the HL7's Clinical Genomics Work Group and the GA4GH Genomic Knowledge Standards Work Stream. Dr. Freeman's research program focuses on developing scalable methods, tools, and infrastructure that facilitate the translation of genomic data to clinical practice with an emphasis on knowledge management and delivery, as well as the development of data and terminology standards. Dr. Ginsberg is the founding director of for the Center of Applied Genomics and Precision Medicine at Duke University Medical Center, and for MEDX, which is a partnership between schools of medicine and engineering to spark and translate innovation. His research addresses the challenges of translating genomics and digital health information into medical practice and the integration of precision medicine into healthcare. He was a member of the advisory council to the director of the NIH and a co-chair of the National Academy's Roundtable on Genomic and Precision Health, and is also a founder and president of the Global Genomic Medicine Collaborative, which is a nonprofit organization aimed at creating international partnerships to advance the implementation of precision medicine. Dr. Freemuth, Dr. Ginsberg, I'll turn everything over to you for the introduction of the panelists.
Thank you very much, Ken. As Ken just described, this session will explore some of the challenges and opportunities related to the integration into and use of genomic data within a fragmented healthcare environment. This figure is from the NHGRI strategic vision paper that was published in Nature last October, and I think it's been shown at least twice already during this meeting. It illustrates the connection between basic genomics research, which generate large data sets from which new knowledge is derived, and the translation of that knowledge into a nascent genomic learning healthcare system, which can build on foundational understandings at the molecular level to enable deeper understanding of the impact of genomics on a patient's health and the utilization of those data for clinical care. For this virtuous cycle to be possible, research is needed to address critical gaps and limitations within clinical electronic systems. Next slide, please. We have an august panel of speakers for this session. Marilyn Ritchie will discuss some strategic opportunities and challenges to using genomic data in a clinical environment. Sandy Aronson will outline a complementary set of barriers to information exchange, and Ken Kawamoto will identify challenges and provide recommendations for scaling solutions through interoperable standards. We will have time for only a short question or two after each presentation, but we will have a more in-depth panel discussion at the end of the session. Our first speaker, if you could bring up her slides, please, is Marilyn Ritchie, who is Director of the Center for Translational Bioinformatics and Associate Director for Bioinformatics in the Institute for Biomedical Informatics at the University of Pennsylvania School of Medicine. And she is the Associate Director for the Penn Center for Precision Medicine. Dr. Ritchie's research focuses on developing novel approaches for understanding the relationship between our genome and human phenotypes. She has an extensive bibliography of more than 350 publications, and she has received several awards and honors, including Fellow of the American College of Medical Informatics. Marilyn, the floor is yours.
Great. Thank you so much for the invitation to participate in this wonderful meeting. Before I get into the meat of what I want to talk about, I feel like I should start by giving you kind of two biases that are kind of part of my personality and my view on this topic that I'm going to talk about. One is that I am an optimist, and so while there are a lot of ways that using our genome for life is challenging and could be problematic, I'm talking about this and thinking about this from an optimistic view, where I think that this is something we should be doing research towards. Obviously, the rest of us do too, or we wouldn't be participating in this meeting. But I'm going to talk about the highlights as to why we should be doing this. And then the other is that some of the points that I'll make are not taking into consideration the regulatory elements around these viewpoints and some of the ethics and really there are nefarious things that one could imagine could be done if certain things happen and we build this infrastructure for our genomic data to be available through our lifetime. I'm putting those things aside because what I want to talk about is the research agenda and the research that we could be doing in this space so that we could move this forward and use our genomic sequence data throughout our lifetime, recognizing fully that there are ethics and regulatory challenges, but that's not what I want to focus on today. What I'm going to focus on is the research that we could do in order to enable this to happen. Next slide, please. So once upon a time, we would use genomic information and genetics in the clinic really in the context of someone presents to a physician with some sort of illness or disorder, and genetic testing was ordered so that a physician could use that genetic information for a diagnosis or to develop a treatment plan. Next slide, please. But what we're moving toward now is a place where our patients have genetic information when they show up in the clinic, whether they've obtained that through a vendor and direct to consumer, and they quite literally walk in with it, a printout report or the data on a thumb drive, or the data are being aggregated because it's being ordered for something somewhere else, and then the data just exists. And so we have the information on patients. And I think we're moving to a place in the future where we could have data on every patient stored somewhere in the cloud. And instead of using it at the time to diagnose, perhaps we could be using it to develop prevention plans for our patients or screening paradigms for the patients. And if they do then present with an illness, we can use that for diagnosis and treatment. When we have this information accessible to the healthcare system, we have the opportunity to identify patients who are at risk of developing certain conditions. We could do deeper evaluations or deeper phenotyping of those patients based on their genetic risks. We certainly could imagine a time where we could identify patients to participate in genotype-guided clinical trials. I think we're seeing a lot of evidence that for particular genotypes, there are medications that work in a better way or a more effective way than other patient populations. So we could do genotype-guided trials if we had access to the genetic information. And then we can also, and we are starting to see that we're returning information to patients about their risks of either developing disease or of adverse drug reactions. And this is the future that in some ways and at some healthcare systems, this future is now, but this is the future that we want to get to. Next slide. There are a number of ways that we can imagine using our genetic information for healthcare throughout our lifetime. Certainly the Mendelian disease risk genes, many of those are used in children and as part of newborn screening. Many of those are also used later in life as we start to develop different later onset conditions that happen in kind of midlife and later in life. That information would be useful to healthcare providers. When a patient presents kind of at midlife with a condition, being able to couple that phenotype with their genetic phenotype to actually make a diagnosis would be incredibly useful. This was a point in some ways that was brought up in the last session in the context of Heidi Ream's question. What if someone did get exome sequencing to look to see if they had a particular variant in, let's say, a cancer gene, and perhaps they didn't have a variant, but perhaps they do have a variant that is important for epilepsy or for a cardiomyopathy or an arrhythmia or Lynch syndrome. Perhaps the testing, they were looking for breast cancer genes, but they identify something for a different type of cancer. But perhaps that information isn't necessarily being returned to the patient or the provider at the time, but years later, as the patient presents with a phenotype, and I think Lisa made this point in the chat, we would want to be able to marry that phenotype with this genetic information that we have. We need to have that data stored in the EHR and accessible in the way that a patient and a provider can understand it. Pharmacogenomics is the one that's most near and dear to my heart. That genetic information is useful when a patient needs to have the drug prescribed, but we would need to have that information in the electronic health record or available to the provider and or the patient in order to use it. And I think where we're heading in the future are these polygenic risk scores. I think there are a few traits where these are being used in the clinic currently, but I think that this is an area that will continue to expand. And if we had the genetic information accessible across all of our care providers, and they could use that to estimate what we are at risk for, and especially as we start to have additional medication, comorbidities, environmental insults throughout our lifetime, those polygenic risk scores will get more and more predictive and be more useful if we have access to them. Next slide. So I think there are a number of opportunities for clinical informatics to conduct research in this space so that this can be a reality. One that is ongoing is the EHR, or electronic health record systems. They are starting to build capacity to store genomic information as structured data. I am not the first person at this meeting who has said this. This has come up before, but this really is key. The variant data has to be structured such that it's machine readable, not just the PDFs that we've done for many years. One example of this is the Epic Genomics module. I should disclose, I guess, I have no interest in Epic. I have no stocks in Epic. That just happens to be the EHR vendor that the University of Pennsylvania is using. Next slide, please. But we've been working very closely with Epic to use the genomics module that they make commercially available in the context of our system, which is called PenChart, by building out this precision medicine tab. So the screenshot that you're seeing, I'm using with approval from Epic. So this is a view of the EHR that you see with the different tabs in our system. So the precision medicine tab is a tab that we've built just over the last couple of years that captures the genomic VAR files, or the variant files, which enable you to have structured content. We've actually built this out with at least one molecular lab. I think we're working on a couple of others right now, but the Ambry testing lab, we have it built out that they can return the results as discrete elements using HL7 directly into our Epic chart. And then genomic indicators are the knowledge, the interpretation of that variant in the context of the clinical data. And then the genomic indicator is what we use to build the clinical decision support. We just published a paper on this in December in Genetics and Medicine that I'm happy to put in the chat when we're done, the URL to that. Next slide, please. So the other opportunities, before I talk about the challenges, with clinical decision support, we can disseminate the relevant information to providers so that they can use it. I think that there's enormous potential in the clinical decision support if we do it well, and specifically for using it outside of medical genetics clinics. The medical geneticists are trained to think about this. So this is that one group of providers who actually do get this content in medical school. Many other providers don't get this, and so we need to train them. One thought is leveraging the CDS systems and leveraging Epic, if we do it well, in order to teach the providers what content is relevant to them. It becomes very important not to have too many alerts, because we all hear about alert fatigue. So we can't alert them to every relevant pathogenic variant in a patient's chart, only the ones that are important in the context of the care that that provider is providing. I think the health information exchanges are another opportunity where relevant genetic information can be shared across health systems. That is one mechanism that information could follow patients wherever they go. I think Sandy's going to talk a little bit more about this in the context of genomics. I will say there are limitations and challenges to that, which I'll talk about in just a few minutes, or in just a minute. And as we get more knowledge, we need to be able to push that information forward. Next slide. So the challenges, there are many. Clinical decision support, we need to build the genomic indicators such that they can be updated regularly, and that the providers have it in a language that they understand. The health information exchanges, currently genetic information is not uniformly shared. Not all systems, especially depending on vendor, can access that information, but we do need a way to leverage these EHRs and have the content kind of passed from system to system, because as was mentioned earlier, some patients get specialty care at one clinic and primary care at another. And then the genome annotations. As we learn more knowledge, how do we make sure that that information is pushed to the people that need it? Next slide. The last challenges, these are actually mentioned from the survey. So many of you answered these specific points. There are compute limitations around the costs of storage and egress from the cloud. That may only exacerbate health disparities if we don't develop approaches and mechanisms that allow us to use this data uniformly, whether you're at a community health system or elsewhere. And then the ontology standards around the phenotype and the genomic annotations. We have lots of standards, but we need to do research that allows us to determine which ones are most adoptable across the different systems. And we have to get away from only the academic medical centers. These have to be adoptable at our rural clinics, at our community health centers. And next slide, which is my last slide. Just to summarize, I do think that there is a lot of positive research we can do by leveraging the vendors and the tools that they're building, but doing the research to figure out how do we get this to the providers in a way that they can understand? How can we get the annotations updated regularly? Informatics can do this. We just need to develop the right paradigm in order to make it happen in a timely way and make it accessible to all. Those health disparity issues will only get worse if we don't find mechanisms and approaches to do this that are low cost and accessible to all. And with that, I will stop and happy to participate in the questions and the discussion later. Thank you.
Thank you very much, Marilyn. Appreciate the talk. We have been taking some notes here. We've got a couple of questions that have come in through the chat, and Jeff and I are teeing some things up for the panel discussion at the end. We do have just a couple of minutes here for a couple of questions before we move on, and there are a couple of things I'd maybe like to highlight. Aaron wrote in the chat here, if you could comment a little bit further on the University of Pennsylvania's implementation here. Does that precision medicine tab that you're building out there work for clinical trials, and to what extent does it support reanalysis?
Oh, that's a great question. So I think the short answer is yes, it could be used for clinical trials. Currently, it's mostly being used in the context of clinical genetic testing. So within that tab, a physician is able to order a test. And so currently, they are ordering mostly from medical genetics clinics, but we have some ongoing research to figure out how do we get non-medical genetic specialty clinics to do those orders. So we are starting to think more about specialty clinics and primary care. And then for the second part of your question, the reanalysis, because the data are structured, they can be used for reanalysis. So we are doing a lot right now to analyze not only the variants that are in there, but the clicks that are happening through that tab to try to learn about how well it's being adopted, who's looking at the results, how are they using those results. And based on that, actually, one of the things that we've just built that just got released this month is once a provider orders a test and the result comes back, instead of only notifying the provider, it's notifying the provider group. And someone from that group goes into kind of this holding place to look at the result, look at the interpretation, and then make a decision to push it into PennChart for patient view. So it'll also go into My Penn Medicine, or defer so that they can have a conversation, between the genetic counselors and the medical geneticists, or to not push that result forward. So perhaps, there wasn't anything conclusive. And so rather than push it into the chart and confuse the patient, they keep it and do additional testing before it gets pushed to the chart. So we're starting to monitor that so that we can do research on it and learn, what's working and what's not working. Right now, as I said, it's mostly being used by medical genetics, although we have seen a few orders from neurology and the hearing loss clinic. It's mostly been medical genetics, but we want to learn how do we get it out of medical genetics clinic. They know what they're doing. We need to get this information to the other providers that need it as well.
That's great. Thank you, Marilyn. Given the time that we have here, I think what I would like to do is reserve the rest of the questions that have been coming in here for the panel discussion. And so we will come back to as many of those as we can. As I mentioned, Jeff and I are carefully trying to record all these, so please keep the questions coming. And we will get back to as many of those as we can. Our second speaker for today is Sandy Aronson, who is the Executive Director of Mass General Brigham Personalized Medicine Information Technology. His teams are focused on both supporting clinical genetic testing and on using technology to improve clinical processes, partnering with clinical leaders to redesign clinical workflows to reduce cost and improve outcomes. Sandy was a founder or co-founder of several companies, including Gene Insight, which provides him with a dual perspective on challenges and solutions for genomic medicine. Sandy?
Thanks, Bob. Can you hear me? Oh, can you hear me?
Yep, you're just fine. Thank you.
Oh, great. So I have really been enjoying this conference, and I hope to add in a small way to some of the concepts that have been brought up while talking about why the heck do we not yet have a broad clinical genetic information exchange? And more importantly, what do we have to do to make sure that when we do have one, it will help rather than harm health equity? So just a quick, disclosure. My employer does get royalties from sales of Gene Insight, which does include some health equity, health information exchange infrastructure. So the dream that many of us have been focused on, I'd say since about 2005, surrounding genomic information exchanges goes something like this. What if all of the clinical laboratories of the world could send structured genetic reports into an information exchange that would route those reports to any of the provider organizations that may order their tests and deliver them in structured form into the patient record, where it would be combined with other clinical structured data, and not only that, but where the world's knowledge repositories would also be integrated into this exchange, so that whenever there's a change in variant knowledge, change in gene knowledge, that would be sent through the exchange, delivered to the provider, hybridized appropriately to the patient record. Those things would come together, and we develop clinical decision support that delivers the power of genetic knowledge right into the point of care, improving care, but also giving us the ability to constantly learn from this combination of genetic data and clinical data, so that as we improve our genetic knowledge, that knowledge will flow back to the genomic repositories, be distributed through the genetic knowledge exchange throughout the world, thus sparking the continuous learning, genetic aware health information system, and delivering the greatest improvements to human health that we've ever seen. That's the dream. So what's the reality? So the reality is, to the best of my knowledge, every one of these exchanges that exists now that lives in the clinical world is small. And why is it small? It's small because every time an institution, a stakeholder, wants to connect to one of these exchanges, there is a cost. There's a cost to set up the connection, and there's a cost to maintain the connection. Vendor infrastructure, standards, best practices can all help a great deal, but there's still always going to be a cost. And the reality is that the number of institutions, if you include physician practices, we're talking about hundreds of thousands of institutions. The only way that we can ever fund this is if those institutions en masse start to decide to invest their own dollars in connecting to these exchanges. So what do they need to believe in order to do that? Well, first of all, they need to believe that it's going to be technically feasible for them to do that. And I think that eMERGE-3 provided a great deal of evidence that, yes, this is technically feasible. Second, they need to believe that this is going to deliver clinical benefit to specific patients, that the decision-making, if they invest in this infrastructure, IT infrastructure, it's actually going to change the decisions that they're going to make, and that's going to improve patients. Lots of work been done on that. I think that, as others have mentioned, I think that the biggest hurdle here is financial viability. That if they make this investment and maintain this investment, that there's going to be some other source of dollars that's going to come in that's going to pay for it. And once they've gotten over all of those hurdles, they have a potentially viable project, but that project needs to be prioritized against every other potentially viable project that is a priority for them. So how does this value proposition for that prioritization lay out? So I think if you look at it for clinical laboratories, I think there's a strong argument, always hard to free up dollars, but a strong argument for the laboratories. It's going to make their tests more powerful for their customers, and it's going to differentiate them relative to any other laboratory that is not on the exchange. What about the knowledge repositories? I'd also argue that knowledge repositories, their fundamental existence is based on disseminating knowledge. So I think there's a strong value proposition for them too. The challenge really lies on, or I think the largest challenge lies on the provider side. And the value proposition for the providers. And perhaps the most important point that I'd like to make is, if we think about this, if there is, a lot of us are from institutions that may have very narrow margins, but have margins and are larger. And we can invest in cutting edge things that don't have an immediate payback. But if you think about institutions that are operating on no margin or negative margin and having to make the decision on whether to invest in this kind of infrastructure, they can only do it if there is an immediate source of dollars that is going to be generated as a result of that investment. So if we can't deliver that value proposition for them, we're not only creating a situation where the patients of that institution won't be served, but we know that often the clinical use of genetics drives a great deal of the research use of genetics. So we're excluding that patient population from certain forms of research and increasing the risk of that research being less equitable. So the cost and like hard economic math of these value propositions, I think actually has a significant impact on equity, as many others have mentioned as well. So what does this value proposition look like for providers? Well, there's cost and there's value. So fundamentally, the cost comes down to, you have to pay for establishing the IT infrastructure, you have to pay for maintaining the IT infrastructure, and you need to pay for establishing and maintaining the clinical processes, the changes to the clinical processes that are going to use that IT infrastructure to actually deliver the value that you're seeking to deliver, the new clinical services or capabilities that you're seeking to deliver. So what does the value proposition look like? So what I find in my role is that the value proposition for people who actually control the spend of clinical dollars, every one of them really wants to do what's best for patients. They really want to consider things like what will reduce the total cost of care for that patient over time. But the actual numbers that they have to make pencil in order to make their budgets work often come down to things like patient acquisition. Will providing this service lead to more patients coming to the institution? And what will the effect be on either fee-for-service revenue or value-based care metrics, which get complex to play against each other? Within our institutions, often grant revenue and things related to thought leadership will come into play as well. But I think that we need to keep in mind that for many institutions, those aren't drivers of dollars, and therefore that this can bias us towards infrastructure that's more accessible for us than for other institutions. So what suggestions can I make, put forth for research? So there are three. What I'd suggest is that it would be really useful to convene forums of the provider economic decision makers to ask them what types of genetic services, what types of genetic use cases would excite them enough such that they would love to invest money in them so that they could provide those services, and what value levers would those services move in order to generate that excitement. And I think that the type of people that we'd really benefit from talking to are people like CEOs, COOs, CFOs, chief medical officers, chief marketing officers, both from large institutions with reasonable margins and also from small institutions, ideally with some representation from some of them with negative margins. And I think that that would help us identify what types of functionality we could focus on, what types of genetic services we could focus on that are most likely to be adopted. And then we can think about what types of research is most likely to give rise to the development of those services and the killer apps that could actually be the apps that would drive people to spend money in order to implement them. And then that would open up the possibility for really exploring, okay, what are the clinical workflow and economic barriers to the IT support that folks most want in order to expand their clinical services and offerings. And this is a place where I think standards can really come into play, where we taking the broad standards that are intended to be generally applicable and really making sure that we make them extremely robust for the specific kinds of apps that we hope to make as cheap as possible for folks to implement. I think that these types of things would be enormously difficult to execute on, but I do think that they could help create a path towards developing a path for creating a much broader and more equitable genetic information exchange infrastructure. Thanks a lot.
Thank you so much, Sandy. Great talk. Thank you for all those ideas. There have been a couple of comments here in the chat and one question by Jeff. Jeff, would you like to chime in on this? Jeff, you're currently muted.
Great. Am I unmuted now?
You're good.
Yeah. Yep. Okay, great. That was a wonderful talk, and my comment in the chat you answered after my comment to some degree, but it really is defining the short and long-term, value propositions, as well as how does this integrate with value-based care. But I wanted to, instead of asking that as a question, I wanted to really endorse your idea of bringing the leadership together that have to make the financial decisions, and also to include both leaders and Luddites. So we have the ones that have actually taken the risk and have shown maybe some ROI to the investment, and ones that don't know where to start and why they should even be paying attention to this. And having that as some of the stakeholders that are going to really receive the value of the research agenda, I think is critically important. So I'm really happy that you raised that, and I hope Mark and Ken will take that forward as a potential action item, and really have a convening that you mentioned.
Yeah, thanks, Jeff. And I do think the fact that the value propositions will be different for different institutions, I think it's been a theme of this conference, and also an important thing for us to consider and make sure we have that diversity in doing that.
Thanks, Sandy. I was struck by your final points regarding the economics, and the way I interpreted it was the need for carrots in addition to sticks to motivate that institutional investment. Do you see any parallel between those challenges and the ones our domain faced regarding the cost of sequencing prior to the development of next-gen sequencing technologies? Do we need a similar revolutionary advance in clinical informatics methods and technologies rather than incremental ones to break through those challenges and get to the point where clinical genomics infrastructure and tools can be widely and cheaply deployed and integrated into clinical environments?
You know, Bob, my take on that is that clearly that would be very helpful, and that would be awesome if it occurred. But I think it's important for us not to focus just on that, because I think that the other side of it is, what are the services that are going to, sort of fit into the world as it is, as opposed to the way we would like it to be, in terms of generating enough value for institutions to offer. And I think that comes down to understanding what kind of genetic services do we think we could enable institutions to offer that they could actually get reimbursed for. And then driving down the cost of those services, I think, becomes critically important. But we need to understand that there's a revenue model in addition to understanding how we control the cost.
Great, thank you. I think there's one more question that came in the chat here. I think I would like to propose it to you, Sandy, and if it's something you'd prefer to defer to the panel discussion at the end, we can do that. It's from Gail. She asks, how does the movement of patients into and out of health systems, depending on things like their job and insurance, affect the value proposition? Are there any potential solutions?
This is critically important, because I think that we often feel like if we can reduce the total cost of care of caring for a patient over their lifetime, that should be attractive to providers. And providers want to deliver that value, but that value does not always accrue to the provider. It doesn't even necessarily accrue to the payer. The dynamics are complicated relative to the payer. So we need to find, and that's where I think we need help understanding what services will generate return in the current environment that will also deliver that kind of value to society.
Great, thanks so much, Sandy. Our final speaker for this session is Ken Kawamoto, who is Associate Chief Medical Information Officer of the University of Utah Health and Vice Chair of Clinical Informatics in the University of Utah Department of Biomedical Informatics. At the University of Utah, Dr. Kawamoto leads the Reimagine EHR initiative, which is a multi-stakeholder effort to enable standards-based interoperable applications and software services to improve health and healthcare. He has developed some very impressive clinical applications and has earned recognition for his work. Dr. Kawamoto is also a member of the U.S. Health IT Advisory Committee. Ken, looking forward to your talk. Take it away, please.
Awesome, thanks. Can folks see my screen?
Yep, looks good.
Excellent. Well, it's really great to be here, and this has been, you know, I think we've been all zoomed out, but this has been actually one of the most interesting meetings I've attended in the past, I guess, year now. So thanks for everyone organizing this. I think it's been great, and it's nice to see so many folks that we haven't seen for a long time. It's kind of crazy, and Jeff was one of my key mentors when I was at Duke doing NHRIK. So it's great to be here. So I'm going to talk a little bit about the standards around FHIR that I think are important and relevant here, and then talk a little bit about some of our experience using these standards in individualized medicine, and touch on opportunities and challenges. I'm going to probably go fairly quickly through this so that there's more time for discussion, but if anything's unclear, please just holler and I'll stop. Just as disclosures, I have honoraria, consulting, sponsored research licensing, or co-development in the past year with Hitachi, Pfizer, RTI, UC San Francisco, MD Aware, and the U.S. Office of National Coordinator for Health IT Aware Security Risk Solutions. I was also, until recently, an unpaid board member of HL7 that develops a lot of these standards I'm going to talk about. I'm an unpaid member of the U.S. Health IT Advisory Committee, and I've helped develop a number of tools which we may commercialize. Okay, so I'm going to start kind of here, right? Like, why are we even talking about standards? And I think it's kind of obvious, but I think these are, at least to me, some of the main reasons. So I think for discovery, there's a lot of value in having normalized data sets for being able to do science on it, right? So I think that's really important. And for clinical care, we need to figure out scalable ways that we can optimize care at the patient level and population level. And the desirable feature of standards is there's an opportunity to say, if we do it this way, we can do it in such a way that it won't work just at our health system or just on the Epic EHR, but it can work on the Cerner EHR, it can work on eClinicalWorks, it can work in federally qualified health centers. So that's the idea and the hope of using standards. And I think what's also important to note is that there's a lot of synergies that can be achieved by using the same standards in the research realm, in the discovery realm, and the clinical application realm, right? Because the data Sandy was mentioning too, like the data is being generated for clinical care, and if we use different standards, it won't talk with each other. So I'm going to just briefly talk through some of the relevant standards. So for a lot of folks, this is probably going to be too basic, but bear with me. I'm just going to make sure we're all on the same level. So FHIR, Fast Healthcare Interoperability Standards, is an HL7 standard. It allows us to exchange health information. I think the key parts here is it's gaining rapid industry adoption, right? We've had standards forever. The key difference, I think, is this one's actually being adopted in health systems and EHR vendors. And it's endorsed by NIH, which is always good for grants purposes, right? Because, you know, NIH says specifically we endorse it. There's several versions, really not too much to worry about there. And what there are things known as IGs or implementation guides. So folks oftentimes don't realize, but like you can use FHIR to, in a standards compliant way, say, my car is a Toyota. It has, 6 cylinders and my wheels are 19 inches and I have the, luxury leather package. Like that is a FHIR compliant, message you can create. Like it's that loose. What allows it to actually be interoperable is what are known as the IGs. And what's most important is, we proliferate a lot of IGs, but the only one currently that is universally adopted and actually is enshrined in regulation as that needs to be adopted is something called the US Score IG. And it's required as a part of this US Score data for interoperability notion. So this is, I would keep in mind that like a key goal should be to get whatever is important to this community into the US CDI if possible. And like I was chatting earlier, like there's nothing genomics related, even in the common stage, right? And we can create all the IGs we want, but if we are the only ones implementing it, it'll have limited impact. Smart on FHIR is related to FHIR. It's basically a way to embed third-party apps into the EHR. This allows, like what Sandy was discussing, as like these killer app notions. And again, the really interesting thing is this has widespread EHR vendor support. So that's key, right? Like there's been a lot of ways to do a lot of similar things. The key thing is vendors are actually adopting this. It also supports patient-facing apps like Apple Health. It uses OAuth 2 to delegate users' data rights to an app. You read and sometimes can write data. And I'll talk a little bit about some other standards that can be used within these to encapsulate your logic. And in some cases, like on the Epic EHR, you can augment the EHR FHIR server to add needed data or to filter out unnecessary data. In our institution, for example, we have about 8 people on our team certified to build new interfaces into Epic. So basically, if you can see it, we can pull it, right? But that's not always going to be the case in other EHR systems. CDS-6 is another FHIR-related standard. It's been a standard since 2019. There's increasing adoption. Epic does adopt this and adopt it pretty well. So you can use this within your rules engine. And it's a companion standard to Smart on FHIR that's meant more for suggesting things. So for example, if you place an order, you can invoke a CDS-6 service through the best practice advisory framework in Epic, and you can retrieve data and provide guidance back like, hey, change them to this, order this medication, order this genetic test, et cetera. There's also something known as the clinical quality language. This has been an HL7 standard since 2015 and is starting to get widespread use, I'd say primarily because CMS is requiring this for their electronic clinical quality measures. And it allows you to do things like express computable phenotypes to support logic and clinical quality measures. So for example, for the eMERGE work in computable phenotypes, it probably, I would think, should be expressed in CQL. The last standard I'll briefly talk about is called FHIR bulk data access, or flat FHIR. So this addresses the issues that most FHIR use cases to date has been a per patient access. And that's just way too slow for population level data retrieval, right? For example, for research purposes. And this is intended to address this issue. And this could allow you, for example, to get normalized large data sets using the standard. The main issue here is this is still in development. It's still early on. I think this could have a breakthrough impact on this field. It's just fairly early. Okay, so with that, I'll transition to just talking a little bit about how we're using this and share some of our experiences at the University of Utah. We started a multi-stakeholder initiative about 5 years ago, I guess, to leverage this and other related standards to try to improve care value. There's institutional investment of about $1 million a year we put into this. So with this, we've been an early leader spanning research and operations. And I think what's been kind of unique here is that we have a really agile, collaborative, innovative environment. Some would call it Wild West, but it's really easy to do things that would take much longer than elsewhere I've learned. And with this and our steering committee co-chaired by our CIO and CMIO, to date, we've deployed over 10 solutions into clinical practice using these. And right now we're a little over $35 million in grant funding. So I'm just going to briefly run through a few examples, which I think kind of illustrate how these things could work. And these are all systems that are in clinical use now at our health system. So this one it uses the Smart on Fire dashboard. It's a collaboration with Hitachi. And what we did was try to address the issue for diabetes. After you give metformin, it's really not clear what to give in most patients, right? It's kind of a coin flip. So what we did is we created predictive models based on our data and the state of Indiana's data, and created pretty good predictive models. And now you can access within the system information like, hey, for this patient, you know, for an A1C goal of 7 in 6 months, if you just continue on metformin on the left-hand side with the blue bar, you can see it's about a third chance that they'll be able to reach goal. If they lost 5% of body weight, it actually doubles it. If you add sulfonylurea, it's about 50%. GLP-1, it's about 80%. But then GLP-1 costs a lot more, and we have insurance-based information that we provide as well, saying, hey, this patient's on Blue Cross Blue Shield, and this is what it looks like for them. Here's another one for lung cancer screening. This is based on ARC R18. And the issue here is low-dose chest CT screening for lung cancer could save more lives than breast cancer screening, but current screening rates are pretty abysmal, and shared decision-making is really important because the individual risk is very different, and it's actually required by CMS for payment. So what we do here is we pull in all the patient's relevant information and provide patient-specific risk assessments that can be actionable, and it looks likely that this is going to end up being in the Epic Foundation system, so then it'll be available to all Epic customers kind of as a natural course. This final one is on a population basis. This is for FHIR-based population health management. The goal here is to identify and manage individuals at elevated risk of breast or colorectal cancer. This is an NCIU 24 where Guilherme Delfio, my colleague, is the contact PI, I'm co-PI, and has led to another NCI Moonshot Initiative grant. But the issue here is, you know, over 10% of us are at elevated risk for these early onset breast colorectal cancer. And, you know, and usually we're unaware, right? Usually just a tragedy that something happens and it's like, oh, well, it's more than that. We have all sorts of data we're capturing in our systems to be able to identify it. So what we're doing here is using a bulk FHIR type approach to gather data on our entire population, running it against CDS services, identifying risk for, and it turns out there's thousands and thousands of patients who are at increased risk that, you know, currently we're doing nothing about. And we're using techniques like chatbots to automate the genetic counseling and genetic testing processes. And, you know, it's quite doable, and I think, you know, it makes sense to do things when we have the data rather than just doing what we currently do, which is generally leave it really hit or miss approach. Okay, so that's just a brief summary of some of the things we've worked on. I'm just going to transition real quick to challenges and some thoughts. So what are some of the key challenges you run into, right? So as with everything, a lot of it's about the data. Data normalization is key, right? Like that doesn't like magically disappear when you start using FHIR. It does in some areas, but in most areas it's like, well, if the data is not clean or data is not mapped, data is not clean, data is not mapped. So that's a key one. Another one that typically a lot of folks underestimate is execution performance. Some of these standards and their implementation by EHR vendors are pretty slow. So for example, in some APIs in Epic, if you want to pull the patient's past medications, like even like hey, asking the question, did they get prescribed something that's getting canceled today? It can take like 10 seconds to pull, right? Obviously, those are huge issues when you're looking at clinical use. EHR vendor support also and standardization is currently mostly just in the basics, right? So, you know, great if it's supported, and if it's not, you know, it's like it doesn't really help you. So these include detailed clinical models. I mean by that things like, well, it's great you can get the office BP, but can you tell how they did it? Was it an AO BP? Well, and you don't automatically get the home BP or the 24-hour BP, those kind of things. There's basically zero genomic support, and at the current state, you know, it's not. It's at least in the way the standardization is going. I think it's a long ways away, and probably something that this community can accelerate. And there's very little in the way of population-based approaches that support in a standards-based manner. So as soon as you move out of patients in clinic, I want to do something with the patient, and more, I want to survey them for risk and recommend, for example, you know, that they get this genetic test. It starts breaking down. And then, of course, the last mile of translating into practice, because this, and especially when it's translating into practice outside of a clinical trial context, because all the things Sandy brought up comes into play, like, well, who's going to pay for it? Why would they do this? Why would they do this rather than buy another MRI machine and run a bunch of scans and get their money back in a year? Those kinds of things. So some recommendations, I'd recommend coordinating and synergizing with the broader health IT community, right? Like, it's just such a common thing in the standards realm in particular. We tend to like to build our own standards and criticize the other standards for like not thinking about us enough. But we'll get beautiful standards, but then nobody will implement them. So I think key here in particular is how can you get the stuff needed for this community into the US Core FHIR implementation guide. I think that should be a central thought in this community if you want it to be widely adopted. We also need to recognize that hard problems largely persist, right? Like, FHIR has gotten wide adoption by promising it's super simple, and it's super simple to create a test application. But if you want to do something real, it's actually still hard. So I think that's a message, right? Like, it can't be like, oh, we have FHIR, like, now it's a totally easy IT issue. That totally is not. And then we need to work together to accelerate key need development. So for example, the US core data for interoperability, some of the key criteria are it's implemented in multiple health systems across multiple EHR platforms, and that will elevate it in the promotion process where it will then come to committees like the committee I'm on that evaluates these to say, should we consider making this a national requirement? Right. And so there's a lot that can be done to basically force the hand of regulators to say, like, hey, at this point, like, this is proven, it needs to be done. And then, of course, we need to facilitate the full translation life cycle, including the last mile of clinical implementation, because I know it tends to kind of go by the wayside in a lot of scientific institutes, but I think, and obviously the whole life cycle is important, but in the end, I think we need what we're developing to be used. Okay, so that's it. And I think I'm actually a little bit over time. So thank you very much.
That's great, Ken. Thank you. We are right at time, but I do have one question that came into the chat that I'd like to toss your direction before we turn it over to the panel discussion. Mark writes, implementation guides are very interesting. Regarding genomic applications, could creation and deposition of an IG be required or strongly suggested as part of the dissemination, implementation, and data sharing plan?
I'd say probably not. I mean, the IG, first, the tooling is kind of painful, and there's only a few people who actually really know how to do it. So it could be really, really frustrating for people who don't know how to do it, right? Like, I believe, at least until recently, it still might be the case. It's like Excel spreadsheet based to build websites. Like, just imagine that for a second, right? I would say no, but I think the notion of, how are you going to, I wouldn't say a requirement, but maybe even just like asking people if it's getting into the applied side, how are you going to use standards? How does it synergize with available standards? Is probably a useful thing to ask. Yeah, the challenge with standards is it's really complicated, it's really deep, and you need really specialized folks who, attend weekly calls and follow all these things and review ballots. And I know Bob is one of those folks, but there has to be a way to scale it because it's oftentimes not possible for a regular, researcher in this field to spend the sufficient time to be truly expert and engaged in that area.
Thank you very much, Ken. I completely concur with your assessment there. It is hyper-specialized and very tricky. I would like now to turn the platform over to Dr. Jeff Ginsburg, who will moderate the panel discussion. Jeff?
Thanks, and that was great. And I can't resist quoting Mark Williams, who has been known to say, the problem with standards is there are so many to choose from, or something like that. But anyway, so yes, we're going to have about 20 minutes for panel discussion. There's already a question from Richard Gibbs for the panel, which is that with various health information exchanges is this a logical place where genomic standards should enter the ecosystem, and how could this happen? And I'll ask as many panelists who are able and willing to respond to these questions to do so.
Go ahead. No, well, it makes sense. I don't know if health information exchanges currently do actually exchange genomic information, but yeah, I mean, seems like a reasonable place to start.
Yeah, I mean, my feeling is it really has to start with the provider side use cases to like define what the genomic information is going to be used for. And then I think the genomic and the health information exchanges can play a really important role in delivering that data.
Yeah, I would agree with that. For our implementation at Penn, we don't currently push genomic information through the health information exchange. So as I mentioned earlier, we're an Epic shop, and so Epic Care Everywhere is the HIE that we're using. We could have turned on genetics, but we didn't. And at least to start with, that was by design, because we're already trying to make sure that the providers that are local know what to do with the information. We don't want to push information out to providers who don't know that it's coming and don't know what to do with it and don't have the interpretation and the content there for them to use it. And that it's partially also, I guess, a liability issue. We don't want to put something in front of a provider that they can't use, but then we've also punted liability that now they have this information that they can't use. So that's something that we're trying to work through. How do we make sure that not only the content is there, but the interpretation and how would a provider that doesn't know anything about genetics know what to do with it?
Okay, thank you. And I want to mention that as I'm going to take the moderator's prerogative and ask that as you answer questions, that you do so with your hat on as advisors to NHGRI and to Mark and to Ken about what the research agenda coming out of this meeting should be. And I'd like to turn back to some of the challenges that Marilyn highlighted in the first talk. We don't have them all captured here, but you listed the dynamic nature of genomic information and annotation, the standards of content and the standards of interpretation, the subsequent use in clinical decision support. So first of all, maybe you could help us, I'm speaking to all of you, help us prioritize the research agenda around those features, and also whether they should be sort of decoupled from each other, or whether they need to be done sort of as one process. If you could begin to unpack that, that would be great.
I'm happy to go first. Can you say one more time the unpacking of which two components? The
fact that we have a dynamically changing genome annotation and interpretation that has to then be put into some standardized format for access by the provider community and for CDS to use. Are those 3 or 4 maybe separate research agendas, or is it really just one? And how do we begin to approach it?
I think it's at least 2 research agendas. At least that's my knee-jerk reaction to it. I think one is what is the content and that needs to be updated, so that annotation information. I think in some ways that is what ClinVar is intended to do. And one of the things that I haven't, and this is my naivete, so someone here may know better than me, how they've done kind of in comparison with something like CPIC, where there are very specific guidelines that go with the annotation and the interpretation. I feel like CPIC has done a really good job of, yes, here are these annotations, and they do evolve over time, but then there's also, here's the interpretation of it, here's what you do about it, what is the action that you take. I know that ACMG has a list of genes that we should focus on, but I'm not sure they have that same level. In PGX, it's a gene-drug pair with guidance. Could we have something similar that, ClinVar has this rich database of annotation? Could we have the annotation, the disease, and the guidance standardized and put into guidelines? And maybe it has been since I've paid attention. It's been a while since I've really dug into the ClinVar data because I'm so focused on PGX right now. But I do think there's that piece, and then there's the CDS that goes with it. I think those are separate people that build out the CDS, certainly. And this is something Carol and I were just talking about in the chat, how you build it in a way that the providers can read it. And so we need to figure out what is the language that they speak, because I think right now we're almost experiencing a language barrier that the informaticians that have expertise in genomics speak a language and we're being driven by the standards that we're trying to use, that's not the speak that the providers use. And so we need to write the decision support in a way that you can understand it. I think that's a little different from the annotation itself. We can have a standard to annotate it, but then we have to build that interpretation engine into the English that clinician speaks.
Yeah, I really agree with Marilyn. And perhaps to say the same thing and coming at it from a slightly different angle, and I recognize that this is a huge generalization that isn't always true, but I do feel that often within the research agenda, the funded research, we're building infrastructure with the intention of enabling 1,000 flowers to bloom. And I think that there's a need for focusing more on specific flowers and driving back into the infrastructure from the flower. So for example, like a specific flower, and I'm not a clinician, so this may not be the right thing, but if there was a service that we wanted to make more broadly available, whereby if a cancer risk variant is found, we want to provide the IT support required to help ensure that that patient over their lifetime gets the necessary screening, and that's updated as our understanding of the implication of that variant is updated. If we were to focus on something like that, then we can drive back into, okay, what do the standards need to be to support that use case? What does the knowledge infrastructure need to be to support that use case? And really try to fund sort of more horizontally or vertically focused than horizontally focused. Which, as an IT person, I love the infrastructure stuff, but I think that sort of those end-to-end use cases are just really important to make it accessible.
So can I?
Thanks, Andy. Sorry, was that who spoke up?
It's me, Ken.
Oh.
So I was wondering, we've talked a lot about the use of FHIR and USCDI code, USCDI for in these 2 days meeting, but one thing that we still are missing is understanding how the FHIR can be used for research. And I was wondering, based off of based off of what we already know about FHIR, where would be a good place for us to start to build evidence that FHIR could be used for research? Because I think that's how you move forward to actually address some of these things as far as transmitting data. But I don't know, I'd like to get feedback from this group, like if you had to come up with a research agenda for this, where should NHGRI when it comes to working with FHIR and USCDI?
Yeah, maybe I'll give it a thought. I mean, obviously there's a whole bunch of research, right? Like if it's clinical research and how you actually, use these in practice, then it can obviously be important there, because probably the first step is you develop the evidence that this is the right way to care for patients, regardless of if you're doing it with, with an EHR or not. Then, figure out like what are all the different ways you can do that. And then it's like, how do you scale it? How do you make it economically viable? On the discovery side, I think the benefit of using something like FHIR is there's a lot of mapping activities occurring within institutions to make the data in FHIR mapped. So for example, there are efforts in probably all of our institutions to map through the Beaker lab mount module the lab components to be mapped to link codes and to be mapped appropriately. Well, if you use the FHIR interface to pull that data, it's going to be done. And a lot of those issues with data mapping for getting data for research and running clinical phenotypes, et cetera, would potentially go away. So I think it's use case driven, but I think you probably just need to get folks together who are trying to solve problems, and kind of like Sandy's notion, right? Like first identify what you're trying to do. Don't start from the technology and the infrastructure, start from the problem you're trying to solve, and then get the relevant folks together and say, well, how could we do this? And then solve it.
One interesting opportunity that may have some good overlap between the clinical and the research realms is cohort selection. So so I think there's an increasing, I think that you might have mentioned this, Ken, and some others over the course of the day. There's, on the clinical side, I think that there's more work being done now to like scan electronic health records for patients who are being suboptimally treated, and develop those cohorts to act on. I think that that's the same kind of work that's often needed on the research side to recruit patients. So that may be an area that could be focused on.
Yeah, I like both of your suggestions, both Ken and Sandy. The one thing that I think it's tricky around the researchers is, and I think somebody said something in the chat, I think Kevin, your comment in the chat just sparked this for me. One of the challenges, so writing a grant on infrastructure to somewhere like NLM, there's innovation, we're gonna build this new widget and it's gonna do all these cool things. Writing the grant on, I'm gonna use FHIR, which is this cool new technology, but somebody else already built it. And now I'm gonna do these like little tiny one-off tests is not innovative. It's part of what we need to do is take this technology and innovation that's been built and test it in use cases. In putting on both my grant writer and a grant reviewer hat, they're gonna get dinged on innovation in terms of, because people typically think of innovation as the cool new thing. It's not a cool new thing. So just something for us to think through. How do we ask these questions in a way that allow us to get at the crux of the issue, recognizing that part of the innovation was the technology that's been built.
So there's a lot of chat on the use case topic coming up. So, and Mark, I think you may have started this, so I'm going to turn it over to you, but I'd like, if possible, to marry the use case idea and agenda to Sandy's emphasis on the economic value proposition, because if we can get a research agenda that actually tells us what to measure and how it will convince the ultimate stakeholders who have to support this, I think it would be a winner. So Mark, I'm going to ask you to elaborate a little bit and see what the panel thinks about, if we can drill down a little deeper on the use case idea.
Sure. I just wanted to, I'll just comment just a bit on Marilyn's last point. I think that, the point that you just made is one that NIH is well aware of. And I know that in a lot of study sections now, we are being told an application of a technology to a new area should be considered innovative, but it's taking a little while for the study sections to kind of pick up on that. But I think that problem is recognized and hopefully will continue to improve. So the use case idea, I think, is really interesting. And the thing that I like about it is in the clinic and in clinical informatics, everything is based around use cases. And use cases can be evaluated and prioritized in terms of where do we invest our resources. So the question then is, could we base a research agenda on specific use cases that could be prioritized by, in this case, NHGRI, and then to get at the point that Jeff was raising, what would be the criteria by which you could prioritize given use cases? And you could imagine, this is where you could engage with a variety of different stakeholders to kind of get their perspective on what would drive their interest in a given use case, whether it's closing a clinical care gap, whether it's an economic argument, whether it's a patient-centered focus, whatever. But if you could actually prioritize use cases, articulate those explicitly, then you might get research that would have a higher likelihood of kind of crossing from the research realm into the implementation realm.
And if I could just add quickly to that, going back to the discussion we had earlier about convening the sort of health system leadership and payers to be part of that conversation, to know sort of what's burning a hole in their wallet that maybe genomics and informatics could solve. That would be, more of a needs-driven approach. We often are, I'm just saying this from my own vantage point, we're often developing technologies that are looking for a solution or a problem to solve, rather. And maybe we need to flip the paradigm a little bit more than we have as a community. So Ken or Marilyn or Sandy, do you want to respond to that?
Yeah, maybe like I've thought about this kind of stuff a lot, and I've engaged in building like our cost accounting system in our health system, which was very interesting, right? Like you you figure out how the money flows, and I think if you get the CEOs, CFOs, et cetera, you need the other, I think somebody said, right? Like, I forget who mentioned, but it was like, you need those folks and the people who are really knowledgeable in this field to say, these are the case studies that show this can be done. Because you get them together and they're just going to say, well, tell me how I'm going to increase revenue or reduce costs. Like that's right. And they're not gonna be like, maybe we can do this. No, like, and then you would bring in the case studies and say, look what we've done at, this amazing academic medical center. And this is how we think it could scale. What do you think? I think it would need to be that combination, or it'd be a very quick kind of conversation.
Yeah, I agree with all of this. I think it would be incredibly exciting to base a research agenda, or at least part of a research agenda, on use cases. And I think Ken's right. You have to seed them. The decision makers aren't going to know what is possible, but they can react to what is the potential economics of what is possible. And I'd also highlight that I've been exposed to these clinical ROI analyses much more recently, and they are intense and complex and take a lot of time to do. And I think look very similar to a lot of things that we attempt to do on the research side. So it may be that there is a research agenda that includes investing in generating those things, because it's not like we can just ask a CEO and have them have them be able to articulate that this will or will not generate a return. I think they can provide guidance, and then we can investigate whether they would create a return.
Yeah, the one thing I would add, having the people from the C-suite at the table also helps broaden the perspective of where do you want to see the return? So one of the things that we've experienced, if you have only the one clinic, and the department chair from that clinic at the table, sometimes they're focused very much on their budget and what ROI would this have in my budget. Whereas if you get the CEO and the CIO and the CMIO, they're thinking about the whole system. And so there are bits of information that we're talking about here that might not help in cardiology, but are absolutely going to help in OB-GYN and in pathology or somewhere else. So definitely that's what we've seen. Having those folks at the table has helped. The other is having researchers and providers and the IT folks all part of the conversation. I mean, that part of the way that this precision medicine tab at Penn happened is that we have a regular meeting that have not only the C-suite folks and the clinical providers and department chairs and the researchers, also legal and bioethics. We meet on a regular basis and talk about these things. And I think to Ken's point, they have to all be part of that conversation to be able to even drive those agendas and infrastructures forward.
Yeah, I would note though, I mean, it's not rocket science to know what you need to move, right? The things that will get CEOs, CFOs to really pay attention is it increases revenue, because like you provide the service in the outpatient, you get paid more than it costs you, right? Like it could be just, you like if you just have that for genetic testing, you get paid more than it costs you, then that would really drive genetic testing, right? Or in the inpatient setting, it reduces your costs because you generally get paid by MSDRGs, which is fixed based on your diagnosis. And the sooner you can get the patient out or the less stuff you do for them, the more you make. It could be even things like other testing you would have otherwise done. Just do it, the day after they get discharged in the outpatient, which could make a several $1,000s difference between doing it inpatient versus outpatient. Like the basics of hospital financing isn't like that super complex. It's just like in the inpatient, generally we get paid a fixed amount, so reduce costs. And outpatient ED, we generally get paid more the more we do, unless you're in a value-based contract. So, figure out ways to increase revenue. And that could be like you do a genetic test and you find that somebody has this like, really complex disorder that requires a lot of treatment. And maybe you just show that, look, like you do this and you get a lot more revenue, and that's enough, that kind of thing.
Yeah, I think that there's many different types of use cases. There's There's functional use cases where you're providing a certain kind of functionality to everyone. And there's use cases that are intended to enable a new kind of service that you're going to offer. And when you're enabling a new kind of service, that's where I think those assumptions about, okay, what additional fee-for-service revenue or impact on value metrics might occur associated with this. And there are trade-offs. Where it really gets complex is, I think that many institutions now have both fee-for-service revenue and value-based revenue. And something that helps one can hurt the other. And you have to play out what this actually means for the institution in that kind of environment. So the different use cases may have different complexity, but in general, I think that the field will get the most value from a research perspective on focusing on use cases that involve enabling new kinds of services.
So thank you for that. I'm going to ask, I did ask Mark if I could ask one quick, well, hopefully quick question, but I know it's a big one. And it's orthogonal to the discussion we're having, but maybe this will come up later on. But Eric Green emphasized the need for a capacity building of the workforce and diversity. And I think the field of IT and bioinformatics and this group, needs to weigh in on how do we actually do that? Because we don't have the pipeline that we need to really enable what we've talked about over the last couple of days, and we certainly don't have a very diverse one. So I'm curious as to whether each of you could have a lightning rod, sort of 15 seconds of thoughts or 30 seconds of thoughts before we close the session.
I believe that in addition to hiring diversity, one of the problems that we have is we don't know how to design for equity relative to supporting the workflows that will actually make these services available to underserved and minority communities. And I think by focusing on that, there's the potential to develop relationships with those communities that then in turn help us with diversity in hiring.
That's a really interesting idea. Thanks, Sandy. Marilyn or Ken, do you have any additional thoughts? Thank you.
Hey, Ken, you look like you're
Oh, no, I think it's always the case, right? We have to start early, but this becomes more just like a societal thing. In the end, we have to start with early childhood education and childcare support and all that kind of stuff. That being said, it is obviously hard. All of us are trying to address this, and it is super hard. Maybe one of the things that a group like NHGRI could do is just take what's happening in every single department, every single program across the country, say in this area, and figure out how can we figure out ways to do that better together. Because it is happening everywhere, right? This is a key issue everyone's trying to address, and it's I'm sure we're wasting enormous amount of cycles rethinking the same thoughts that somebody else already knew.
Yeah, and I would say what I keep coming back to in my mind is how to use IT and informatics to reach people where they are. And, can we, so certainly on the provider side, can we push content to them that they can understand? On perhaps the, undergraduate side, we have all this content. Can we just learn from other educators? How do we take the content that we have and write it in a language that they can understand and then deliver it in the platforms they use? Perhaps we should be making more educational videos on TikTok. That's what the kids are doing these days. So can you take these concepts and meet them where they are in the language that they're speaking?
Wonderful. It was worth going over 3 minutes and 35 seconds to hear those important thoughts. And I want to thank Ken and Marilyn and Sandy for outstanding contributions, and also Bob, my co-chair, and helping us move the session forward. Now I'm going to turn it back over to Mark and to Ken. Thank you.
Thank you for a wonderful panel discussion. So this concludes session 5. We now have an hour's break, so to give Mark and I some time to kind of pull all this information together for the next session. So we will reconvene at 3:50. And thank you again. Mark, is there anything you want to add?
Nope, but please come back and ready to discuss. There are a lot of interesting themes. We don't have a lot of time, but we'll try and make sure that we have captured all the important discussions. So thanks very much.
Thanks. Mark, I'll set up a separate Zoom meeting for us. Give me one moment.
Okay. Bob Freemuth wrote and asked if I was going to go into the hurry up offense, and I think it's going to be more akin to watching Patrick Mahomes trying to escape from the Tampa Bay Buccaneers front four. So in any case, what's going to happen for this next hour is that we're going to go through some slides that Ken and I put together with the takeaways. Yeah, Tampa Bay didn't take it easy on Mahomes. Hoffman, why should I? So Ken is going to be editing slides with the content as we go through. We do have a lot to cover, so I'm going to be fairly draconian about moving us forward, and would ask that if there are some minor comments or edits, pop those into the chat or the Q&A. We will definitely pick those up. But if there's some major themes that we either got wrong or missed, that's where we really want to make sure we don't leave anything behind here. So Ken, are you ready to share your slides? Ken was feverishly preparing the slides, so he will probably be joining shortly.
I'm here.
Here we go. Okay, great. Thank you, Ken. So just to remind people of the goal of this meeting was to develop a research strategy on the use of genomic-based clinical informatics resources to improve the detection, treatment, and reporting of genetic disorders in clinical settings. So we want to make sure and focus our comments around research strategy, research questions, as opposed to doing what we all too commonly do, which is to get hung up in some of the issues relating to infrastructure and standards and that. Next slide, please, Ken. As I listened over the course of the 2 days, I pulled out what I would consider to be overarching themes that came up several times and were not specific to a session. And so these are the overarching themes that that Ken and I heard. Research should include an implementation science framework. We need to work on understanding the value proposition for all stakeholders, which would include patients, research participants, providers, payers, the C-suite, researchers, et cetera. And to that end, the NHGRI should support, potentially convene multi-stakeholder collaborations to explore this value proposition to make sure that the research is relevant. We need to have an ongoing engagement with patients as partners to develop applications that are under patient control to promote genomic medicine. And in particular, to identify ways to lower barriers related to regulatory processes to promote research in this area. One of the ongoing challenges with consumer-focused research is that the private sector can do this very rapidly, but whenever we try and do this under a research umbrella, we frequently get bogged down in a lot of regulatory issues with IRBs and consents and third parties, et cetera. So are there ways that we can accomplish what needs to be accomplished with this patient engagement and still do it in a timely fashion? One of the things that I think was most striking right from the get-go are the pervasive bias across all aspects. And I think we initially started with the idea that there's bias in our genomic databases, but I think we heard many speakers talk about bias in the data, in our information systems, in access, in definition of value and knowledge, and probably a number of other things. So I think we have to recognize the pervasive nature of the bias and make sure that we explore that up front. And then the last here is the need to account for relevant workflows, that everything that we talk about impacts somebody's workflow We need to understand that, and we need to research it from the perspective of the workflow. Pat DeVerka added that we need to develop and utilize core outcome measures to demonstrate value. I think that's an important point. So we'll be sure to capture that. Jeff had indicated our goals are aligned with the private sector. Why not include them in some way? I think that's what we're trying to get at with the multi-stakeholder collaborations, we will make sure and expand on that. Any other comments on this particular slide?
Yeah, Mark.
Go ahead.
Yeah, just on implementation science, one thing I've observed is two very different interpretations of what that means. When I talk with some of my friends in behavioral and psychology research, they take that to mean evaluating the impact of a behavioral intervention. I think in the EHR world, we see implementation science as how systems are implemented. So as we get behind that, we'll want to be really clear on what model of implementation science we're referring to.
Yeah, and the NIH actually has a dissemination and implementation group that works across the NIH on this, and they have some definitions. And so I was sort of implicitly assuming that we would use definitions from the NIH around this, but that's a good point. And a good implementation science framework should specify all of the different answers to those questions. So that's a good point to consider. Any other questions on this particular slide?
Mark, I just put into chat a comment about the last point on workflow. We have heard a number of different times how genomic data is disrupting the practice in some ways because it is providing opportunities for clinicians and other providers to think in new ways about how they practice and what data points might be relevant in a particular situation. So existing workflows may not be readily geared towards building in the consideration of genomic data. That might present an opportunity for us to help develop new workflows around these data types, specifically rethinking the situations in which it might be most advantageous to start considering genomic data for a particular patient, and think about how the introduction of those data in those new workflows might be a goal that we can work towards in the future, help shifting the clinical practice into those new workflows, as opposed to trying to shoehorn an extra step into an existing workflow.
Okay, there are a couple more comments that are coming up in the chat, but to make sure that we get through everything, I'm gonna make sure we will capture those. None of them, I think, they're pretty self-explanatory, so I don't think we would need a clarification or discussion. So Ken, let's go ahead and move to the next slide. This is just, I'm not going to really have any discussion on this slide. This is just a summary from the survey that I presented yesterday. The key needs from the survey: research into methods for integrating analytical interpretations derived by computational models of genomic data into clinical settings, and then study ways to ensure that CDS has the ability to incorporate and support multiple genes in clinical information. These are a little bit more generic than some of the deeper discussions that we've had. And then a couple of the other things here really support some of the overarching themes that we've noted on the prior slide. So let's move to session 1. So session 1, we heard loud and clear that we have to study the inherent biases in data, algorithms, information systems, and in implementation. These are multidisciplinary, multidimensional, and this is certainly not an exhaustive list, but ones that were explicitly discussed were race and ethnicity, social determinants of health, urban, rural, academic, non-academic centers. I think education could fit in here as well. I thought there was a really interesting comment that was made about implementation equity. I think Kevin Johnson mentioned that. That was a term that was new to me, but is really hit home. So that's one that I took away there. A second bullet, and I have a second slide on session 1 here. So the need to explore the value proposition, which is imbalance between researchers and participants, and this is particularly noticeable in underserved populations. Ken, let's go to the next slide. We'll go through the bullets and then we'll come back for discussion. We need engagement in research across a broader range of organizations. We have a Casablanca problem, round up the usual suspects. NIH puts out an RFA and we see all the same faces, most of which are also on this call. And again, this concept of implementation equity. We need innovative research questions and methods to address these inherent biases in a rigorous and systematic way. This was a really interesting point that Dr. Jeff brought up. And we really don't have methods right now that we can roll out to implement this. So this is going to require some research into actual methods. And then outcomes have to capture both benefits and harms. This was specifically mentioned in conjunction with genomic clinical decision support to inform some mitigation approaches. So across those bullets, any additional comments? Jeff had mentioned about literacy and numeracy. I'm going to say that's another part of the list, and we have to be make sure that we're covering that. Rex mentioned a few, yes, we will include more detail from the Desiderata and survey in the final report, but I didn't want to spend too much time in the discussion on that at present, unless there's a specific point that you wanted to bring up. Okay. Any other comments or reaction to the takeaways from session 1? Okay, Rex can't unmute. Can somebody unmute Rex?
Rex, you should be able to talk now.
Can you hear me now?
Yes.
Ah, excellent. So there were just a couple of things that I thought actually resonated with some of the other things we heard in later sessions. So one of the comments was we needed to do a better job capturing conditions across multiple genes. And I think given the broader interest in polygenic risk scores, that's something that probably should get added. Again, I'm just not sure what level you want to deal with these things. And then we did talk a little bit about in that session, we talked about the apparent inconsistency between people wanting to be able to track the actual details of the molecular test / analysis and wanting to make and on one hand, but on the other hand, wanting to make sure there wasn't too much detail in the EHR. So I think those need to show up at some point, but.
Great. Okay, and then could somebody unmute Suzanne Bakken, please?
Sorry, I don't need to be unmuted. My chat is just disabled. I want to be able to chat.
Oh, okay. So, and if you want to plop it into the Q&A, so we should be able to capture it there as well. But yeah, if we can at this point essentially enable chat for everyone, that would be great. Gerald, I don't know if you can do that or.
Yes, that has been enabled. So
excellent.
They can do that now.
Everybody can chat.
Yeah, and
okay.
I wanted to add something, if that's okay.
Yes.
The little hand raise. Oops. So I was just wanting to add that the learning health system and enabling the learning health system from genomics, because this was just in the example with eMERGE, it enables discovery and clinical care, but that loop was not necessarily closed completely. And so that's something that could be added as a bullet.
Great, thank you. That's really important because we did see that figure from the strategic plan several times during different presentations. I might, and we don't have to do it now, Ken, but I might move that actually to an overarching theme, is the whole learning healthcare system, because I think that figure would frame that discussion very well. Other reaction to the takeaways from session 1?
Mark, this is Erin. I would just add for the implementation equity that I think this is going to take some outreach on our behalf. I think when these, certain organizations, when you feel inferior over a long period of time, it's hard to be courageous and step up to the plate and try to go after these interesting opportunities and so forth. So I think we, as a group too, we need to do outreach to those types of organizations that maybe are kind of missing from this group.
Very good point. All right. Hearing no other comments and not seeing anything else appearing in the chat, let's move on to session 2. Again, 2 slides on the takeaways here. Use of implementation science research methods to improve implementation equity, not just research equity. There have been a number of comments that have been coming in around implementation science frameworks. And so we're getting some good engagement around that. And I think we'll be able to address some of the definitional issues that have come up as we prepare the final report on this. Develop a patient-centered research agenda. There were a number of potential research questions that came up in that session. Authentication, privacy, security of genomic data that's successful or under patient control, recontact, absent clinician oversight, a knowledge requirement for different stakeholders, sort of what is that minimum knowledge set to be able to use the information, innovative enabling platforms for obtaining and returning genetic results, and the ability of a patient-centered focus to potentially reduce bias. And the next slide on session 2, Ken. Need an updated business model of research to attract a broad range of stakeholders to participate, to understand more about the incentives to implement genomic-based clinical informatics resources and tools, and research into ways to represent genomic information as structured data while minimizing manual processes. That also comes up a bit later from the discussion today. So we're now open for discussion on session 2. Comments, questions, additional points to make.
I raised my hand, but I don't think you saw it.
No, I'm not seeing the hand raise function, unfortunately. So
yeah, I can see it, so I can call it. Sorry.
Okay, Ken, why don't you take care of that then?
Yeah.
I just put it down, but I do still have a comment. Sorry. Regarding this session, I think something that I didn't emphasize enough in the 10 minutes I was given, but I think is very important, is that we need to figure out certain methods to use to close that loop and do the learning healthcare system that was mentioned earlier. Reusing genetic data that's from test results presents some methodological challenges, and you need to modify the strategy compared to when you use research genomes. In some ways, it's a parallel problem to starting to do phenotyping in the EHR as opposed to having all your your subjects come in for an exam or something like that. So I think that figuring out methods for this reuse model would be really helpful in this case, because some of the limitation of using genetic data in the clinic is because we don't know the full phenotypic implications of some of these variants. I think it's really going to be a very powerful place for us to learn about that.
Yeah, and I think that that ties in with what we heard in today's session from Marilyn about the use of this data over the lifespan. So that reuse question is a really a critically important one, possibly even rising to the level of an overarching theme, that these genomic data are different from most other types of clinical laboratory data that we use on a daily basis.
Go ahead, Robert.
Yeah, thanks, Mark. I'd like to pick up on that point on the previous slide, looking at the patient-centered research agenda. One of the things that came up a couple of different times, so I'm not sure where you'd want to capture this, is the concept of data portability. I think it might fit into this topic as well, but again, it was mentioned a few different places. So being able to access the data, break this, I think, in some ways, kind of a legacy mindset of the data living in only one place, or under the auspices of a healthcare system, as opposed to potentially with the patient. Some research as to how we can improve the portability and access to genomic data that has relevance across a patient's lifespan.
Yeah, I think that that's a really excellent research question. It was something that I didn't explicate under the patient-centeredness, but as I said in my comments yesterday, right now, at least in the United States, the patient is the only common actor across the healthcare delivery system. And so if we have data that is relevant to the patient, that data has to move with the patient. And so that's a really key point that I think could lead to some very interesting research questions. So Ken, as you're taking notes there, if you could just add, making sure the data moves with the patient.
Mark,
go ahead.
Mark, I think another thing that came up in session 2 that I'm not really sure how to frame it in a research context, but scalability of some of the examples that were given, that was brought up several times. And I think that it would be important to capture that in the summary.
Thanks, Carol. Yeah, scalability, sustainability, these are ongoing issues for a lot of what we do. We can be successful over the course of a 4-year grant, but then it dies there, and we need to make sure that we build in some aspects of this scalability, generalizability, sustainability. So thank you for that. Other comments? And I know we're going through this very rapidly, and one thing I've learned in my decades of doing this is that not everybody processes information at the same speed. This is really trying to react while we're inundating you with information. So these slides will be available, and I would encourage those of you who would like more time to process this to feel free to take that time and respond to Ken and myself asynchronously, and we'll make sure we incorporate that as we pull everything together, because we're certainly not going to have the report on this meeting ready by next week.
So Mark, quick comment on the scalability and sustainability piece. If I'm not mistaken, in eMERGE, at least in the early phases of eMERGE, algorithms were developed in one system and then tested in an independent system to be valid. And I'm just wondering if that same paradigm should be repeated here as part of the research agenda, that if we're developing systems that are just unique to a particular place, it's not going to go very far. But if we can show generalizability and port them to other systems and demonstrate similar results, that would be very powerful.
Yes, and of course that was an inherent feature of the network, where it was a requirement to be part of the network that you had to be able to do this. And so in some ways that was baked into the RFA for eMERGE. Now, that's not typically something that, we require dissemination plans and data sharing plans as part of investigator-initiated research. But how we might be able to take some of the imperative to have generalizability and get away from just sort of a generic, well, we're going to publish some papers and put it in GitHub, so that we can actually have substantive examples of generalizability, I think would be a very interesting thing for NHGRI to consider. Okay, let's move on to session 3 then. Takeaways from session 3, and again, I think I've got 2 slides here. Informatics research for genomic evidence computing and genomic knowledge-based construction to enable scalable, shareable, computable inferences of genomic knowledge and harmonization of practice guidelines. There's a lot to unpack there, but I think it emphasizes some of the things that we were just discussing. Research into novel workflows that diminish burden for primary care providers, tap into other healthcare workers and engage patients, and don't default to alerts and reminders. So essentially innovation in the idea of how we actually present this information in a clinical workflow. Studies to ensure that new technologies don't exacerbate health disparities. And ideally, actually reduce health disparities might be a more positive way to frame that. Educational policy research agenda to reduce barriers and improve knowledge for patients and providers. So in other words, research into educational strategies and policy implications that may raise barriers for implementation could be a potential research agenda for NHGRI in this space. Next slide. I stole this from Chenhua Wang because it was so beautiful. So the blue text are my modest efforts to the one that she presented in her talk. I just love this idea of the socio-technical strategies for success. Informatics research for genomic, that looks very familiar to what I just read, so I'm not going to read it again. Harmonize the interests of multiple stakeholders to facilitate team science and implementation science. Incentivize collaborations to foster research on a learning health system for genomics. What is new evidence? Who is affected? What needs to be done? And clinician and patient-centered design of workflow. Thank you, Chenhua, for providing that. So comments about the takeaways from session 3. So Bob noted in the chat that, as Bob always notes in every setting, that this depends on data representation and standards, which is absolutely true. The question then is there a research agenda on data representation standards? And we do get into this a little bit in sessions 4 and 5. About ways that we can actually do research around data representation standards, as opposed to say what we always say, which is we need standards around this area. And then there certainly is a research agenda around knowledge delivery of the complex data for decision making.
Mark, just on that first point, what I was trying to get at is the first point on the previous slide mentioned research into genomic knowledge bases and specifically the construction of them. My point there was constructing new knowledge bases ad hoc with their own localized, specialized models will actually hinder interoperability. And so the new knowledge bases that we are looking for here really should be pre-harmonized and standards compliant whenever possible.
Great, thank you, Bob. Also, two people have mentioned on the last sub-bullet here, more human factors research. And then Terry, you had your hand up.
Sure, I just wanted to make the point on your previous slide, you talked about making sure that new technologies don't exacerbate health disparities. And we hear this a lot, and it's a very important point to make, but it seems to me that clinical informatics could be key in reducing health disparities that we currently have in genomics in medicine. So why not try to be a little more proactive about that?
Yeah, and when I read through that, I tried to fix it on the fly, which is to say, change that from rather than try not to exacerbate to actually reduce health disparities. And I think the other point you made in the chat was that, these disparities exist across many more areas. I captured this one specific to session 3, but in the overarching goals, I tried to be a little bit more global about the idea that we really need to be looking at these types of disparities and biases in all areas that fall under this this topic area. Pat DeVerka mentions economic benefits. Again, I think we're trying to capture that in the overarching goals around value, that by having different stakeholders, we can also accomplish that. But certainly there are the NHGRI is already funding some economic evaluation research under the auspices of the ELSI program. So that would be important. And then fair principles were mentioned, which is something that should be incorporated. And then I see, Guilherme, you have your hand up. So go ahead.
Yeah, on the health disparities issue, I think one point to consider is the technologies that we expect patients to use in certain interventions. It's important to keep in mind, for example, that I think there's Pew research showing that among the underserved populations in the US, 25% of the people do not have access to the internet or a smartphone. They only have a text and voice. Oftentimes we come up with cool things like chatbots and patient portals, and there's a large percentage of that population that won't have access.
Yep. Yeah, I didn't explicitly include that in a sub-bullet, but certainly that digital divide, technological divide is an important thing to consider in health equity and disparity.
Yeah, another point is insurance coverage. We had some barriers in trying to implement some of our interventions with underserved because, for example, if they test positive for a BRCA, uninsured won't be able to cover costs of aggressive cancer screening like breast MRIs.
Yeah, and that's where we get into the real. I guess I'll use the term, even though I really don't like the term, the slippery slope in terms of where can you actually sort of impact in a research program, as opposed to acknowledging the idea that we need to identify how the things that we think are going to improve care could actually unintentionally exacerbate some of these disparities. And so the need to capture exactly that type of information to say, hey, we facilitated all this stuff relating to identify people at risk, but in our healthcare system, a lot of people don't have access to the care that they need to actually act on the information that we're providing, in which case have we really done them a service. Other comments regarding to session 3? Some really good discussion here.
So just
go ahead.
Just on one point that more research involving other modalities for decision support one of the things that Guillermo brought up was chatbots, for example. And so there are a lot of other modalities that could be explored in novel ways.
Yep. Okay. And Chenhua, I just noticed you had your hand up, so go ahead.
Yeah, so I also think we need a policy in place regarding who should be responsible for delivering and acting on this highly relevant or clinically meaningful genomic results, because I feel there's still a lot of confusion there.
Yes, and you've done a beautiful job of allowing me to segue into session 4, because that's in fact one of the takeaways that we captured from session 4. So why don't we go ahead and move on there for given the time. Session 4, we had some takeaways, research on what constitutes a minimum data set for clinical care and research. I love the phrase, I think it was Suba that said, learn from less data. That's sort of a paraphrase of the perfect is the enemy of good enough. Oh, what happened? There we go. Thank you, Ken. Then research at the interface of human cognition and artificial intelligence. How can we take the best of both? Include research into explainable AI to promote clinical implementation. And then research into the development and implementation of a common semantic framework to reduce reliance on manual curation. And do we have another takeaway slide there on 4, Ken? Yes. Research into data interoperability between clinical systems that is focused on the implementation of genomic medicine. I thought a very powerful theme that came out of session 4 was a research based on specific use cases to support genomic medicine implementation through informatics, but to engage with the diverse stakeholders who were referenced earlier to prioritize which use cases really have the most resonance with the diverse stakeholders, so that we can make sure we're doing research on the most important questions and not have something that is ends up being cool but not relevant.
So Mark,
yes.
Mark, I made a mistake. Let me switch something. That's actually,
I was going to say that seemed different to me, but I was just going with, okay, here we go.
I appreciate you going with the flow, but yeah, this is actually the remainder of session 4.
Yeah, okay.
And then the next slide would be session 5. So there, this is where the remainder for session.
Okay, great. So this is where the point that Chenhua had made. Research into the development of a responsibility model across the EHR for patient access to needed information, and also to capture what Chenhua said, which is understanding, who within the healthcare system has responsibility. Now, I did have a question. It came up in the chat, and it was raised about the idea of using 80-20 use cases in genomic informatics, and we didn't have a chance to discuss that during the session. And I was wondering if and Ken, remind me, who had posted that first?
Maybe I'm wrong, but Lisa, was this you who posted that, or maybe I'm wrong in that?
If somebody wants to claim responsibility for that, I'd love to understand a little bit more about how you're thinking 80-20 use cases could be useful.
I think I posted that, Mark. That was in the context of HL7 standards development, comparing HL7 version 3 with FHIR. Yeah, the idea is to identify the cases that cover most of the practical, readily available clinical genomics, things that we could do today, and that could be covered by 20% of the standards.
Gotcha. Yeah, so this is the thing that we frequently end up with, is that we develop a strategy to address a problem, and we can take care of 80% of the problem, but we delay ourselves because we spend a lot of time consumed about the edge cases that don't necessarily fit, whereas we could potentially move forward on the 80%, get that to work, and then go back and try and pick up the edge cases as best we can. Is that a fair restatement?
Yeah.
Okay, I like that. Thanks. Okay, other comments or questions about session 4?
Mark, this is Nephi. I just wanted to make sure that somewhere, I'm not sure is the best way to address this, but to better develop regulatory frameworks to open up the use of genetic data. And I'm not sure exactly how that fits into the research world, but that's sort of a need that needs to be addressed, I think.
Yeah, I think that we mentioned that in the prior session, but it can be refined here, is that it is within the purview of NHGRI, and they have certainly done this before, to research regulatory frameworks and how they either facilitate or in more cases impair implementation. And so that type of research would certainly be within the realms of consideration for Genome. I just, for balance's sake, Ken, in your comment, just say how they facilitate and impair research. Because one might assume that there are, GINA could arguably be said to maybe facilitate, although there would certainly be people who would argue with me on that point.
Yeah, this is Terry. I think it's not only the how they may facilitate or impair research. It's how they would affect clinical care, and maybe affect is a more benign way of saying this. But certainly, I think we've seen with pharmacogenetics that the reports that we get now that basically give you genotypes and nothing else are meaningless to patients and clinicians.
Right. So that's a really good point. So affect research and clinical care, yeah. And Chenhua just made a point in the chat that when we use AI, we may be referring to it more as augmented intelligence rather than artificial intelligence. That would certainly be an important distinction to make as we throw around that term. Other comments or questions about session 4?
Do you want to say something about AI and its potential to exacerbate disparities?
Yeah, I think we can definitely fold that into the point that we made previously about technology, and just make sure that when we think about technology, that we also include things like AI, machine learning, et cetera. Again, this gets it back.
Whatever the difference is between those two words.
Well, yeah, it depends on what you're trying to get funded, I think, for the most part. But it also relates to the point that was made in session 1 about inherent biases in algorithms, that they don't in fact represent unbiased or naive approaches to data. And there was a point made, Casey made a point about that Gil had raised in his talk, smart contracts that have both policy and technical implications, and that's one way to address a responsibility model.
And Mark, I have my hand raised if you.
Okay, I didn't know if it was raised from before or if you had another comment. So
somebody
go ahead.
For me, because I can't figure out how to do that. So you had made an interesting point about synergizing human and artificial or augmented intelligence, and there could be an interesting research agenda around how to do that in genomic informatics. I don't know what it would be, but it might be, and I'm not sure if it's this session or the next session that it belongs in.
Yeah, I know I captured something. I may not have been stated so obviously. Go to the prior slide 10, Ken, I think.
Oh, the interface of cognition and artificial.
Yes, yeah, that was the one, right? And
yeah,
again,
sorry, I missed it.
Well, geez, I don't know how we've been going through it at the speed of a FedEx guy or the, the side effects of medication ad.
Right.
All right.
You might make
And
it
then
a little bit clearer because, interface of human cognition, artificial intelligence. But anyway.
Yeah. Okay. So we'll make sure and just flag that to clarify that some. Chenwa, I'm going to get to you in just a second. Jeff Ginsburg also mentioned that telegenomics was, we certainly didn't mention all of the different technologies that are potentially available. I would certainly think that telehealth, telegenetics would be within the realm of technology augmentation for the research agenda, but we'll make sure that that's included. Chenwa?
Yeah, so I just want to add a little more about that responsibility model, because I feel a lot in genomic medicine, a lot of delayed diagnosis or misdiagnosis are actually due to the poor coordination between the primary care and then a geneticist, the specialist. So I'm thinking, in clinical informatics, there has been a long time researching care coordination. I'm actually thinking, like for genomic medicine, we need a better coordination among all these different type of care providers between specialists or primary care. I think that needs to, that's actually critical. If we can have better support between primary care and a specialist or geneticist or genetic counselor, we can potentially shorten the time for diagnosis and have better patient management. Yeah, I just want to add that.
Yeah, that's a very important point, and certainly is an area of critical importance to informatics research. And as we think about our rare disease patients in particular, this is a major issue. I think it was the Undiagnosed Diseases Network that noted that a review of medical records alone resulted in a diagnosis in somewhere between 5 and 10% of the patients that were referred to the UDN. In other words, the information about the diagnosis was there, but it just had never been communicated to the primary care physician, to the patient and their family. So a very critical point to make sure that we would include that into research agendas. We talk about data and interoperability and all the technical things, that there are these human factors related to handoffs and coordination communication that also have to be considered. Okay, let's move on to session 5. The takeaways here, research into data interoperability between clinical systems that is focused on the implementation of genomic medicine. Develop research on specific use cases to support genomic medicine implementation through informatics, and prioritize those use cases based off diverse stakeholder input. Coordinate and synergize research findings with the broader health IT community. In other words, try and make sure that research findings, research standards that are developed are shared with groups like the US Core, HL7, GA4GH, et cetera, and then facilitate the last mile of clinical implementation, identify what's ready, and support implementation research around it. Other takeaways from session 5.
Hey, Mark, this is Jeff. This may not rise to the level of the kinds of things that are on this slide, but I did want to reemphasize Sandy's call to action with the leaders of systems to convene to be the end users of our research and to help us make sure that our research agenda is moving towards their needs, the C-suite people.
Yes, I explicitly listed the C-suite people on the overarching theme slide. So,
okay, sorry.
It's okay. Again, we're going through this quickly, so it's easy to miss stuff. And I didn't capitalize the C in C-suite, so easy to miss. Either we did a phenomenal job of capturing it or we have completely exhausted the group. Oh, I can always count on Bob. So go ahead, Bob.
Mark, could you say something a little bit more about point 3 there? Coordinate and synergize research findings.
So this was this was the point that came out of our discussion around implementation guides. I think Ken was talking about how we generate certain things in the course of projects that we do, but we don't necessarily pass off the knowledge around some of the standards into the standards organizations. And so I raised the question about, would it be appropriate for the NHGRI to say, hey, and by the way, if you're going to be doing this types of research, you should be creating implementation guides as part of a dissemination plan. And you and Ken thought that was not a great idea because the standards world is a pretty arcane group of specialists. But I think there still is the need to make sure that new things that come out of research that have implications for standards somehow get passed to that specialized community so that they can take advantage of the knowledge of the work that's been done. So that's what I was trying to capture there.
Okay, so thank you for clarifying that. I certainly agree with what you just said there. I didn't derive all of that from the bullet point, but I like what you're saying, and I would support adding some semantic around that. That basically helps to close the loop between the research use cases, the development of standards and the support of standards for those use cases, and then the downstream adoption and utilization of those standards for the next set of research.
Yeah, and as you said that, Bob, it really, it emphasized to me that this is a two-way street. So in other words, the research group that's working on a specific use case needs to make sure that they are aware of and use any extant standards that already exist rather than creating new standards that aren't needed. So that's the front end piece, but on the back end, they may find some gaps in the existing standards that need to be addressed that they can fill, and then that needs to be handed back. So that's a little virtuous cycle in the standards world that should be reflected.
That's right. And in fact, I have a slide on exactly that. That standards development is not linear, it is iterative, it is circular for the pattern that you just described there.
Great. So if you wanted to fire that to Ken and me, who knows? It may just end up in the summary somehow. So, Marilyn, I saw your hand up.
Yeah, thank you, Mark. One point that it came up earlier, but it came up in this session as well, that I wonder in bullet 2, in talking about the use cases, I wonder if being explicit about some of those use cases being the reuse of the genomic information, where, for example, an exome for a Mendelian gets reused for a pharmacogenomic, or a panel for pharmacogenomic gets reused for a PRS or a Mendelian gene, just so that we don't end up siloed, that we have implementation use cases that are in each of those spaces, but we haven't actually done the use case of reuse or integration across those different types of genomic medicine content.
Great. That's a good point that we can definitely add. And then Jeff added in the chat that we did see that learning healthcare system diagram. The left-hand side of that diagram is the basic science. And so even though this workshop was focused on clinical implementation of genomic medicine through informatics, we don't want to forget the idea that, sort of a superset of what we were just talking about in the standards world is that there's going to be additional knowledge that is generated by the implementation of genomics that's going to generate new questions that are relevant to the basic science community. So how can we create that virtuous cycle to make sure that that information gets back into the hands of basic science researchers to facilitate their work? So it wasn't something that we really had a specific time set aside to discuss, but I think if we're going to use that diagram as one of the anchors for our report, we definitely need to say this is an important piece that should also be addressed. Other comments or questions? Okay, we are on time. This was fantastic. Obviously now your job is mostly done. The group that's the organizers will have a lot of things to sift through to kind of pull this together. The next steps, obviously all of the presentations, the video and everything will be posted to the Genomic Medicine meeting site. So that will all be available to anyone who's interested. So if there are people that you think are interested that weren't able to attend, please point them in that direction. We will be looking to generate a final report for the group that we will circulate for additional feedback. That will be used for planning purposes within the NHGRI. However, we're also looking forward to developing this as a manuscript for publication. The challenging thing with these manuscripts is that most journals are not looking specifically for a meetings proceedings type of publication. And so we'll need to reframe the information that we pulled away into more of a research type of article. Those of us that have been involved in these meetings have done that more than a few times. We generally offer the opportunity for authorship to the group that was involved in organizing the committee and also to the presenters. So we'll circulate any manuscript around to the group. The expectation would be is if you want to be an author that you would need to have a substantial contribution, which at this point would include a critical review and comment and approval of the final manuscript before submission. And there are a couple of you who are on the invite list, and I don't know if you're still on the call or not, who actually serve in the role of on editorial boards. And so if any of you are thinking, gosh, this would be a great article for my journal, please let us know, because we would love to submit this to a journal that would have specific interest. We're thinking the primary journal that we'd like to go after would be a high-profile informatics journal, since that was the primary focus of this meeting. But we're open to other alternatives that people might suggest. That is it from my perspective. Ken, any other final actions or closeouts before we call it a day?
Other than I just want to say thank you to the Coordinating Center, Pamela Williams, Taiji, and NHGRI AV people for pulling this all together for us. Thank you so much. This would not have been as successful without your input and hard work. And to our analysts also, Madison, Anna, and Lori for also helping capture notes and helping with some of the logistics. So thank you so much.
Yeah, thank you, Ken, for mentioning that. As big and complicated as a meeting as this was, it was about as seamless from a technological perspective as I can recall. After doing a year of this, that's really delightful. And again, I want to thank all of you who were co-moderators, presenters, and most importantly, participants for all your input. That made the meeting really worthwhile. And special thanks to Ken for he's been involved in planning for this meeting on and off for what, about 2 years, Ken?
Yeah, and some of the co-moderators and speakers were also with me on this. So this has been almost a 2-year effort to get this going. So thank you so much for all of you putting up with me to help get this forward.
Very good. Well, enjoy the rest of your week, and we look forward to hearing from you down the road. So, and at one point, we'll actually be able to see each other.
One more thing, Pamela, how do
in 3 dimensions?
how do you want people to access the slides? Is there a process you want them to use?
Yes, actually, I was just getting ready to chat that out. You guys can email me. You guys all have my email address, I know, because I hounded you guys for it like a month and a half. You guys are probably going to call the cops on me, but so you can just email me and I will share that particular folder with you, or invite you to share that, view that folder, and that should give you everything you need.
So Pam, I was wondering, instead of, would it just be easier to just send to all the, because we have the email list of everybody, could we just
Yeah, and I mean, I could try sending them all, I mean, unless they're asking for just one, but they're very large files, and I'm not sure that you know, Duke will allow me to do that. I know I've gotten a couple of slides from people that they've had to send it to me via Dropbox or something because it was too large to send. So I definitely can try that, Ken. That's, you know, that's a very good idea.
I defer to your judgment. That's fine.
No, I wouldn't do that if I were you, but okay.
Well, that's it.
All right.
We can also, this is Teji. I will also say that, as in the past, the NHGRI website for this meeting historically puts the PowerPoints and the presentations, they link them to that website, so we can also direct people there. Yeah, that might take a couple of weeks though.
Okay. Yeah, and I do have to get with Macaulay anyway, and I can discuss that with him. So maybe he, you know, can give me a timeline. But yeah, thank you, Teji. We'll definitely figure it out.
All right. We'll see you all.
Thanks, everyone.
Thank you. Have a good weekend, everyone.
Thanks for doing this.
Yep. Thanks. Bye.